Great. Welcome, everybody. I'm the country representative for ICA in Canada, also the executive secretary for Poste Norte, Jean-Charles Lavallée, based in Ottawa. We're joined by our coordinator, Dr. Camino Paricio. Based in Mexico City. And we have the task force with us, led by Dr. Amandeep in Canada, and Yasmin Rivera. She's a doctor based at USDA. And Jaime from Mexico City. He's with INIFAP, and we're delighted to host him and his colleagues in their whole group in Mexico City attending today. We had over 300 registrations, so we're delighted to host you for this webinar. Very special webinar. We do this once a year. At Costa Norte, we've been around since 1998. We've been doing these events on a yearly basis on animal health, on plant health, genetic resources. We do a lot of work around resilience in agriculture, so soil health, air, water conservation. The one today is focused on RNA interference tech. And so we're looking at this application in plant health and recent advances. Our focus is usually the North, so Canada, US, Mexico, who will make up our board of directors. And the goal of this workshop in particular for the next 3 hours is to discuss the latest research advancements in this tech for managing pests. Now, the idea is to enhance plant health by selectively, say, silencing harmful genes in pathogens anywhere from like viruses to fungi, nematodes, insects, and the the tech can be applied to plants through different types of expression, like transgenic expression, topical applications, integrated with nanotech to improve the delivery and stability and that. Pretty cool stuff. We The way it's organized is we're using Wordly. So it's AI interpretation. You'll find there's a delay. Most of us are used to having human interpreters, and so you'll see that it's different. It's not word per word. It waits for like the whole sentence to provide some kind of meaning to this. So then you'll see it's not that long, the delay. We're talking a couple of seconds. Any questions regarding that can be posted to the chat. Our colleague Camino in Mexico City is monitoring the chat, can help guide users today in the workshop. Yeah, in terms of housekeeping, the next 3 hours, I'm going to pass it over to the task force lead, Dr. Deep. Then we'll start with two different sessions and a break in the middle. In terms of Q&A, Dr. River will manage the first session, and we'll take the Q&As at the end of the presentations, but don't wait till the Q&A to start asking questions. Post them to the chat. That's ideal because it's fresh. And then the Sometimes Scientist will answer you directly in the chat. And then if they're not answered, then Dr. Rivera can then use those during the facilitation during her session, and Jaime Dr. Mina can do that in the second session. So nothing more from me in terms of housekeeping. I mean, you know the Wi-Fi, you know where the bathrooms are. I usually do this on site, but it's online. So I just invite Dr. Deep to then provide some opening remarks, and we'll pass it to Dr. Rivera after that. Cheers.
Thank you so much, Jesse, and hello, everyone. On behalf of the Proceed Norde's Plant Health Task Force, I want to thank you all for joining us from Canada, US, Mexico, and beyond for the seminar today. And as Jesse mentioned, it has been organized by the Plant Health Task Force in collaboration with ICA participation, and we look forward to a very productive session today. And as JC mentioned, the goal for today's seminar is to focus on RNAi for plant health. And over the course of this workshop, you will hear from leading experts who will share the latest research finding, innovative applications, and the perspectives from across North America. And I encourage you to take full advantage of today's discussion, not only to learn from the presentation, but also to ask questions in the chat and exchange ideas, and maybe consider these scientific knowledge to further utilize within your field of science as well. And without taking much time, I again also want to thank you, our speakers in advance as well, for their time and expertise. And I look forward to the productive and inspiring session. And now I will pass on to my colleague, Dr. Yasmin Rivera from US, who will be chairing our first session. So Yasmin, over to you.
Thank you so much. Welcome everybody, and thank you for joining this webinar on a workshop on RNAi technologies for plant health. We're going to get started. We have 6 amazing speakers, experts from the US, Canada, and Mexico, and we're going to start with Dr. Steve Robinson from Agriculture and Agri-Food Canada. He's going to be talking about developing RNAi-based fungicides to control Fusarium head blight. As a reminder, please put your questions in the chat. If there are questions for a specific speaker, please note that in the chat as well, and we'll do our best to answer them. Dr. Steve, welcome. Thank you for being a speaker, and you have the floor.
All right. Well, good morning, everybody. And welcome from Saskatoon. I'd like to start by thanking the organizers for the opportunity to present some of our work that we've been doing in the range of RNAi. We've been targeting FHB in wheat, and so the next few minutes I'll introduce you to the work that we've started. Okay, so what are we trying to achieve? So FHB is a worldwide problem in wheat. It's of particular concern to Canada, where it's on producers' minds every year. But when the conditions are right, the effects can be devastating. And so not only does it result in yield loss, it can result in mycotoxin contamination of the grain, which can, once it reaches certain thresholds, render the yield worthless. And so what we're trying to do is establish a program where we exploit RNAi and apply it to those diseases where there's a dearth of effective resistance genes. And so in that way, we can complement the work that is ongoing by the wheat breeders. And so together we can develop a solution with an integrated sort of disease management package for wheat producers. As you know, RNAi is not specific to particular organisms, but it does lend itself well to providing strategies to those diseases without clear R genes. And so examples would be Fusarium head blight in wheat, Sclerotinia sclerotic in canola. And so what we're trying to do then is develop RNAi and exploit it to develop what we would be terming next generation fungicides using the specificity that is encoded in the RNAi process. We can target pathogen genes and leave beneficial organisms, and the pesticide would not be toxic to people. And so that is the idea in a nutshell. So, I mean, this is an RNAi meeting, so I won't dwell too much on the processes which underlie it. But in effect, what we're doing is exploiting post-transcriptional gene silencing. And so in our approach, we make double-stranded RNA molecules and we spray these onto wheat heads. Once the double-stranded RNA is taken up by the pathogen, so it's ingested, these molecules get recognized by evolutionary conserved complexes, and so they get recognized and digested into siRNAs. And these small interfering RNAs are created by the process of an enzyme named dicer. The resulting siRNAs are recognized and bound by Argonaute proteins to form a RISC complex, and it's this RISC complex that uses the sequence specified in the siRNA to guide it to find mRNA with homologous sequences. Once this is recognized, the Argonaute cleaves the mRNA target and it causes gene silencing. And so if you can reduce the level of mRNA, you can reduce the level of protein, and if that protein level falls below a certain threshold, you can see a phenotype. And so that's the logic behind using RNAi. We envisaged a strategy which can be separated into 4 phases. So phase 1, you generate sort of genomics data, sequence data of the organisms involved. You use transcription studies to get to know the enemy, and then you can use these genes to design triggers to trigger the RNA process. And then you would screen these in a phase 2 strategy. And so this is sort of back and forth process between stage 1 and stage 2, where you're looking to find the most efficacious targets and the most efficacious triggers to those targets. Once you find targets which work, you might then move to stage 3, where you would test them in the field. And so that then requires engagement with the regulator, the pesticide regulator in Canada. That's Health Canada in the PMRA office. And then upon successful field trials, then that would lead to phase 4, which might lead to commercialization opportunities of the technology developed. So that's the process that we're trying to tackle. Once you've got your list of targets from your phase 1 studies, you can then design your trigger molecules. And so we use a comprehensive sort of bioinformatics analyses where we find the genes of interest. We try to make sure that we're looking at low copy genes where possible. And we make sure that we've got specificity to the pathogen and no off-target effects to key species. We then use these molecules to then clone and trigger to make the double-stranded RNA. And this is all documented, and we can provide comprehensive analyses to the regulators should these molecules make it that far along the development process. Once you have your molecules in hand, you then have to determine which ones are the ones that you want to pursue. And so we set up a process where we screen these molecules. And so what we do is in the greenhouse, we sow out wheat plants every week. And so that there's a constant supply of wheat heads available for testing. So then once the wheat heads reach anthesis, we've then purified double-stranded RNA from our clones that we keep in E. coli, and we've quantified this, and we spray the double-stranded RNA onto the wheat heads. We then let that dry, and then 24 hours later, we provide a point inoculation, and we do these type 2 FHB testing assays. Since it's so dry in Saskatchewan, we add a bag over the wheat heads for 2 days to encourage disease development. We then remove the bags, and we monitor disease progression over the next 3 weeks. And so what we're trying to determine is the effect of the treatment on disease. And so that can occur directly, or most likely it can occur through the process of gene expression. And so the treatment would silence the genes, and the gene expression being silenced would result in a differential disease phenotype. We don't measure disease, we don't measure the gene expression during the screening process. We only do that once we've confirmed that we've got a valuable phenotype. Each treatment is combined of a different RNA species at a particular concentration, and so these are all measured. We then measure the amount of spores that we apply to each wheat head, and then the plants are irrigated, but again, we don't measure the level of irrigation during this process. And so this is the model of the experimental design that we have. And we use a ordinal regression statistical analysis to determine any differences that we observe. And the reason why we use an ordinal regression model is that in order to go through the number of heads quite rapidly, we categorize the disease into using a 6-scale ordinal scale. And so where there's no disease, we score it to 0. Where the disease is located to the point of inoculation, we rate it as a 1. Where the disease is spread to adjacent florets on the wheat head, we rate that as a 2. Where it's gone beyond that, up to 50% of the head, we rate that as a 3. Where it goes beyond that to 80%, it's a 4. And beyond 80%, we score it as a 5. So then we end up with a distribution of data from the treatments, and then we make these comparisons. And since the experiments are small because we're limited by space in the greenhouse, we use a Bayesian method to integrate these data. And so where we see promising results, We use results from the previous experiment as priors. We then generate new evidence from an additional experiment and combine them together using Bayes' theorem to generate a posterior distribution. And all of the information that you need to do the statistical analysis is included in these posterior distributions. So what do these posterior distributions look like? Well, you can plot them, and you can see that you get a mean and a variance for each treatment. And so what we're looking at here is when a particular RNA molecule has no effect. And so when we compare it to a water control, you can see that the mean is about 4 in each case. And when we take the delta, the 0 position on that x-axis lies within the distribution. And so when we see that, we can determine that there's no significant difference. And the same goes with an effect size. And so, as you can imagine, in any screening exercise, many of the targets and many of the triggers that are designed fall into these kind of categories and have no effect on disease rating. But of course, there wouldn't be a story without some success. And so if you keep at it and you keep doing the analyses, you can find particular molecules which are target to essential genes where you see a reduction in the disease. And so in this case, it's quite clear. You almost don't need any statistical analysis. The distributions from water and the double-stranded RNA treatment are clearly different. When you run the analyses and you calculate the delta, you can see that the 0 level is way out on the far from the distribution, the posterior distribution of the deltas, and the same with effect size. And here we've got an effect size of 1, which is a large effect. And so just to sort of remind you where that lies on the ordinal rating scale, we've dropped the ratings from an average of about 4 to an average a little over 2. And so we think that that's a reasonable reduction. And then we would like to know, how do these treatments hold up in the field? Because although the greenhouse experiments are very valuable, in order to be effective, we need to deliver this treatment into the field. And so in order to take that to the field, what we need to do is to engage with the Canadian regulators. So that's PMRA, as I mentioned earlier. And so you make your applications, and it takes about 6 to 9 months to have these applications evaluated. They provide you with a research permit. And so we set up trials over the last 3 years at 4 locations throughout Canada, 2 in Saskatchewan, 1 in Manitoba, and 1 in Quebec. And then you're at the vagaries of the weather. And so in Canada, it gets hot and in the summer, and sometimes you have to negotiate the smoke from the forest fires, and you can have normal conditions or drought conditions. And over the last few years, we've been suffering from drought conditions, which is rather unfortunate. And so this then lends itself to what is going on in the field. And so we set up the experiments where we essentially have the same underlying statistical design, but of course now, when we're trying to measure the effect of treatment on disease, we now no longer control the irrigation. And so when it rains, it rains. We have to irrigate the plants in order to establish good disease levels in the disease nurseries. And so they get applied regardless of the timing of the treatment. And so you're at the mercy of when it rains or when the nurseries require irrigation, when you've delivered your treatment. And the same with the spore levels. We no longer control the spore concentrations that are applied, nor the timing of the spores. And so what we end up with is rather than a randomized control experiment that we would do in the greenhouse, we end up with like a sort of observational experiment that we have in the field. And so this complicates analyses. We can wrap up all these differences by location, and then we use a model based on a beta regression model, because we're measuring percentage data in the field with from our collaborators who run the disease nurseries. When we get these data and you run the beta regression models, the coefficients are difficult to interpret when you just look at the numbers. And so We're using a hierarchical model because we're nesting treatments within genotypes and in particular locations. Um, and so just looking at these numbers it's difficult to see what's going on, although you can see the treatments where we do observe statistical significant differences. And so the way that we do this is to then we push new data through these models to see what's going on. And so as an example for treatment ES181, when you push the data through the regression model, you end up with these new distributions. And so you can see that in each case, at each location, we're getting a reduction in the disease rating at each location in each year. And so this is very encouraging, and we need more field years to make sure this holds up because we've been doing these data in dry years. And so I'll just stop here by saying what we're going to do in our next steps. So we need more field trials to determine the efficiency of the molecules that we're examining. We're optimizing the spray delivery because that is an issue as well. We're trying to determine additional genes which will target the mycotoxin content, and then we're looking for a industrial partner to commercialize this technology. And so I just want to acknowledge all of the people involved. And so Mohammed and Trisha do most of this work in the lab, and they've been helped by an array of summer students, and then the collaborators who have done the DON analysis, and then I'm particularly grateful to people like Maria Antonia, Oscar Molina, Samia, Randy, and Sylvia who run the disease nurseries. And with that, I'll thank you for your time.
Right. Thank you so much, Dr. Robinson, for keeping us on time with the schedule. Our next speaker is Dr. Mayra Herrera. She comes from the National Institute for Forestry, Agricultural, and Livestock Research in Mexico. And she's going to be presenting on hormonal shifts in Capsicum annuum induced by Fusarium inoculation, auxin dynamics, and plant growth responses. So with that, I'll give the floor to Dr. Mayra.
Thank you so very much for the introduction. First of all, I want to say that I'm grateful that you were willing to give me this opportunity. And for now on, I'm going to switch to Spanish, if there's no problem with that. ¿Me pueden decir si ven mi pantalla, por favor?
Sí, la podemos ver.
Perfecto. Muchas gracias. Bien, antes de comenzar la presentación, quiero comentarles que todo lo que estaré presentando hoy es el resultado de trabajo de más de una persona. Me representa a mí, pero también a mis compañeros, algunos colegas de aquí del instituto, la doctora Silvia Salas Muñoz, el doctor Luis Roberto Reveles, el maestro en ciencias José Ángel Cid Ríos, todos compañeros del INIFAP CESAC. Y por otro lado tenemos la participación de la doctora Fátima Berenice Salazar y Jorge Armando Mauricio de la Universidad Autónoma de Zacatecas, quienes han aportado valioso trabajo para obtener estos resultados. Bien, voy a comenzar. Estoy teniendo problemas. Ok. Voy a comenzar mencionando o definiendo a las auxinas. ¿Qué son las auxinas? Estamos hablando de hormonas reguladoras clave de casi todos los aspectos del desarrollo vegetal, incluyendo la morfogénesis y las respuestas adaptativas. Cuando hablamos de la síntesis de auxinas, pensamos o sabemos que están relacionadas con el crecimiento vegetal, la respuesta al estrés, a cualquier tipo de estrés, ya sea biótico o abiótico, el desarrollo de la planta y el mantenimiento de las células meristemales, entre otros factores, otros efectos que puede tener. Bien, cuando hablamos de una planta que tiene una llegada del sol directa, por ejemplo, hablando de la luz solar, tenemos una distribución uniforme de las moléculas de auxina por todos lados en la planta. Sin embargo, bueno, cuando la planta recibe luz de un lado, comienza una elongación celular. Y cuando el sol incide precisamente de lado, las auxinas se distribuyen hacia la sombra, lo que induce la forma esta elongada de las células y esto también podemos relacionarlo con el hecho de que las auxinas están relacionadas con la forma que empiezan a tomar las plantas. Habiendo hablado un poco y de manera muy breve acerca de las auxinas, les quiero platicar hoy sobre la evaluación de cepas de fusarium como hongos promotores de crecimiento. Esto se hizo en el cultivo de Capsicum annum, que es el chile. Aquí en Zacatecas producimos chile destinado a secado. Entonces, todos estos experimentos se realizaron con el chile tipo mirasol. Bien, voy a comenzar mencionando que se trabajó con diferentes cepas de fusarium. Estas fueron aisladas de cultivo de maíz, de sea maíz. Resultó muy interesante porque no se esperaba, vamos, fue como accidental. Las doctoras Silvia y Fátima tenían una colaboración con otra doctora de otro campo experimental y mandaron muestras de maíz para su proyecto, era sobre maíz, pero la curiosidad de las doctoras las llevó a encontrar todo esto, ¿no? Un montón de cepas de fusarium. Algunas fueron catalogadas como cepas benéficas, otras fueron catalogadas como cepas patogénicas. Y después de esto, bueno, pues ocurrió ¿qué sigue, no? ¿Qué más podemos hacer? Todo esto ellas en primera instancia lo podemos observar ahí en las fotografías, lo hicieron en cajas de Petri con Arabidopsis thaliana y después de técnicas moleculares y de secuenciación se logró hacer una identificación de las cepas que se lograron aislar del maíz. Después de esta agrupación de las cepas, se procedió a analizar en plantas de Arabidopsis thaliana la expresión del gen RH6. Claro, esto aparte de muchas determinaciones que se hicieron, el día de hoy les vengo a presentar esta expresión de este gen en particular. Em, ya que, bueno, las auxinas estimulan la producción de ROS y eh, de diferentes especies reactivas de oxígeno y más adelante vamos a hablar un poquito del tema. Ahí les quiero mencionar, por ejemplo, de las que se seleccionaron para trabajar con Chile fueron dos cepas de Fusarium equiseti y una cepa de Fusarium botti, que son las que estoy yo resaltando en verde y en azul. De aquí en adelante es como yo las tengo ya como catalogadas con ese código para cada una de las cepas que se utilizaron. Y bueno, ahí les muestro también los primers que fueron utilizados para realizar la expresión del gen. Em, bien. Ok. ¿Y por qué es importante esto? ¿Cuál es la importancia del metabolismo de las auxinas y la expresión de este gen? Eh, las auxinas, como les había comentado, estimulan la producción de especies reactivas de oxígeno y de óxido nítrico. Estas especies reactivas de oxígeno y el óxido nítrico inciden en modificaciones oxidativas o nitrocitación en auxinas que modifican la expresión de FBOX2 y AFB2 y en el inhibidor de transporte 1, que es el TIR1. Al facilitar la translocación de estos factores de transcripción al núcleo de la célula, esto modula la expresión local de algunos genes sensibles a la auxina. Y la oxidación de estos factores de transcripción, por otro lado, inducirían la degradación de la auxina y la IAA por medio del proteasoma, es decir, se ubiquitina, y se libera la ARF, que es el factor de respuesta a las auxinas. Y esto, bueno, va a inducir cambios en la expresión precisamente de este gen del R de HD6 y de RSL4, que también es un gen importante, que es el Root Herd Defective 6-like-4. Y bueno, a pesar de que nosotros tenemos la expresión de este gen por efecto de estos hongos, Siguen habiendo muchas cuestiones al respecto. Todavía en el 2024, yo hago referencia a un artículo que obtuve del 2024. Existen algunas preguntas abiertas que quedan, ¿no? Y ahí las señalo en la presentación. ¿Cuáles son los péptidos RALF involucrados bajo estimulación con auxina? ¿Cómo regula la interconexión entre peróxido de hidrógeno y las el óxido nítrico, perdón, los procesos oxidativos de TIR1 y AFB2 y el subsiguiente programa de expresión génica? ¿Operan otros procesos biológicos bajo la regulación de estos componentes de señalización? ¿De qué manera las señales ambientales, incluyendo las fluctuaciones de temperatura, la disponibilidad de nutrientes o las interacciones microbianas, como es el caso de este proyecto, tienen el potencial de promover o inhibir la activación de la vía RAF-FER-TIR AFB2? Se acumulan niveles elevados de ácido 2-oxiindol 3 acético durante el crecimiento de pelos radiculares en respuesta a la estimulación con auxinas y a los altos niveles de ROS. Todas estas son incógnitas aún por las que podemos seguir trabajando. De hecho, nosotros tenemos planes a futuro todavía con este trabajo. Quiero compartir con ustedes que esto empezó desde el 2018, desde que se aislaron estas cepas de Fusarium. Y uno normalmente cuando escucha Fusarium, o era mi caso anteriormente, pensaba en un hongo fitopatogénico, algo que va a producir una pudrición de raíz y que me va a llevar a la pérdida de mi cultivo. Por ejemplo, yo trabajo principalmente aquí en Zacatecas con el cultivo de frijol y cuando yo veía un cultivo con señas de pudrición de raíz, verla de manera aérea, pero saber que era síntomas de pudrición de raíz, lo primero que uno piensa es o es Rhizoctonia o es Fusarium, ¿no? Entonces, pensar en cepas de Fusarium que pudieran promover el crecimiento de la planta era un poco en un principio para mí de manera personal entenderlo, pero con el tiempo he aprendido a sorprenderme, quizá, por decirlo de una manera. Y en este caso, como ya lo mencionamos, en base a los trabajos realizados en Arabidopsis thaliana, se tomó la decisión de seleccionar las cepas que ya les mencioné, las primeras dos haciendo referencia a la M102T y la M102R, a Fusarium xeti, y la segunda, que es perdón, la tercera, que es la P119 a Fusarium boti. Bien. Se realizaron en cámaras de crecimiento algunas pruebas con Arabidopsis thaliana, como ya se mencionó, y en estas fotografías podemos ver más claramente el efecto que tenía estos hongos en la parte radicular de las plantas. Tenemos en primera instancia un control negativo, el cual no fue inoculado con ninguna de las cepas, y por otro lado las cepas seleccionadas, las cuales vemos un desarrollo radicular y radículas secundarias más desarrolladas en comparación con el control negativo. Ahora, cuando vimos la expresión del gen del R, de RHD6, pues no eran necesariamente las cepas que promovían mayor expresión de este gen, pero es importante también entender que no nada más nos vamos a ir por la expresión de un solo gen sin ver otras cuestiones, ¿no? Por ejemplo, no lo presenté aquí, pero también se evaluó la concentración de clorofila y fue más elevada por algunos de estos, el P119, por ejemplo, eleva la concentración de clorofila a mayores niveles y algunos también compuestos de respuesta al estrés, como algunos compuestos de la vía de los fenilpropanoides, que también son más elevados por efecto de estas cepas. Bien, una vez seleccionadas estas cepas en Arabidopsis, se pasó al trabajo con Capsicum annum. Esto son fotografías representativas del trabajo que se hizo en cámaras de crecimiento, en primer lugar, y podemos ver ahí el desarrollo tanto aéreo como de la raíz de las plantas control o de la planta que representa el grupo control y las plantas de tratamiento con las diferentes cepas. Podemos observar de manera general que las cepas tienen un efecto más importante en el desarrollo de la planta que lo que serían las plantas que no fueron inoculadas. Bien, sabemos que, bueno, no nos podemos quedar con el trabajo que realizamos en laboratorio, ¿no? En las cámaras de crecimiento, porque no necesariamente refleja lo que ocurre en la realidad y lo que va a ocurrir en campo. Nosotros realizamos la plantación de las plantas de chile Mirasol en parcelas experimentales en el INIFAP Campo Experimental Zacatecas. Ahí les muestro algunas fotografías de los colaboradores. Hubo bastantes estudiantes involucrados en este trabajo. Y bien, les muestro ahora fotografías de muestreos destructivos que realizábamos en la parcela, en donde, por ejemplo, en el caso de esta diapositiva, les muestro fotografías de plantas que representan a cada parcela. Esto lo teníamos en bloques al azar con 4 repeticiones. Y este primer muestreo destructivo se realizó a los 60 días después de la inoculación de las plantas de Capsicum annum con las diferentes cepas de Fusarium. Tenemos fotografías tanto de la planta completa como del número de frutos que se obtuvieron a partir de estas plantas tratadas. Y podemos observar que la cantidad de chiles, perdón, que la cantidad de chiles, ups, listo, es menor en general en las plantas control. Cada una de esas 4 fotografías, creo que se entiende la dinámica, representa los chiles que cortamos de cada una de estas plantas que están en la parte superior. Y podemos ir observando desde el principio que, por ejemplo, las cepas de Fusarium equiseti fueron más sobresalientes en cuanto al número de chiles por planta. Posteriormente realizamos el muestreo destructivo a los 90 días después de la inoculación. Nuevamente la misma dinámica, tenemos 4 fotografías que cada planta va a representar cada una de las parcelas de las repeticiones que teníamos en campo y bueno, también las fotografías ahí del número de chiles que obtuvimos en las plantas inoculadas. En este caso resalta ya a los 90 días después de la inoculación el efecto que teníamos con una de las cepas de Fusarium equiseti y la cepa de Fusarium botti, en donde vemos un número de chiles, de frutos, más elevado a los 90 días después de la inoculación. Bien, aquí les muestro ya los resultados graficados, ya con el análisis estadístico. Y bueno, además del número de frutos, cuantificamos el número de botones y el número de flores. Y podemos observar que en general el número de flores no vemos una respuesta con diferencias significativas. Sin embargo, en el número de botones y en el número de frutos, vemos que el efecto desde aquí va siendo un poco más diferenciado dependiendo del tratamiento que la planta recibía. Bien, en cuanto a la concentración de las toxinas, me pareció interesante mostrarles un análisis factorial de los resultados, ya que nosotros tenemos un factor que si bien es la cepa con 4 niveles considerando el control, tenemos el otro factor que es el tejido, el tipo de tejido que se analizó de la planta con 4 niveles también, siendo raíz, tallo, hoja y flor. Y bueno, siempre se me enseñó que cuando realizamos un experimento con más de un factor, lo correcto es analizar si tengo yo un efecto significativo de los efectos de interacción, lo correcto es analizar eso, pero me pareció interesante por mostrar también los efectos principales porque considero que tenemos que estar conscientes de qué es lo que estamos buscando. Yo, por ejemplo, comparando nada más cepas o comparando nada más el tejido, puedo observar que existe diferencia significativa con 2 factores. En cambio, bueno, también en los factores de interacción se puede observar claramente un efecto de interacción significativo. En general, las hojas fueron las que tuvieron la mayor concentración o mayor síntesis de auxinas totales, fueron cuantificadas totales. Y aquí en esta gráfica se puede observar un poco más lo que yo les mostraba en las tablas anteriores. Como les comento, la hoja en general siempre tuvo la mayor concentración de auxina seguida de la flor, pero podemos observar que en la flor no hay un efecto tan importante por el tipo de tratamiento. Incluso no lo veo comparando los tratamientos con el control negativo que no recibió ninguno de los tratamientos. Sin embargo, viendo la gráfica superior, el cuadrante superior, podemos ver ahí dónde está la interacción. Me parece completamente interesante que al comparar la concentración de auxinas entre el tejido foliar, si bien el tratamiento 2, que es la línea azul, es la que tiene mayor concentración de auxinas en hoja, es decir, una de las Fusarium equiseti, cuando realizamos el análisis en la raíz, podemos observar que el efecto no lleva la misma tendencia. Si bien en hoja fue esa cepa de Fusarium equiseti la que permitió la mayor concentración de auxinas, fue la que permitió el peor contenido de auxinas en la raíz. Lo que quiere decir que, bueno, pues dependiendo de la cepa va a ser el efecto que también depende de los niveles del factor tejido, ¿no? El efecto es diferenciado dependiendo de qué parte de la planta se está analizando. Bien, y hablábamos entonces hace unos momentos sobre la relación que existe entre el estrés oxidativo con las auxinas. Poco se tiene documentado, no hay mucha información al respecto, pero de manera generalizada podemos mencionar que, bueno, una planta de manera normal, tiene diferentes sitios de producción de especies reactivas de oxígeno. Entre las más, o la más importante en las células vegetales son los cloroplastos, pero entre las más importantes también podemos mencionar los procesos metabólicos que se llevan a cabo en la mitocondria o la NADPH oxidasa. Todo esto, bueno, pues está asociado con la producción de estas especies reactivas de oxígeno, diferentes especies reactivas de oxígeno. Y esto va a modificar la vía de las MAP sinasas o de las sinasas dependientes de ciclina. ¿Qué va a pasar con esto? Esto va a perjudicar de manera ya sea positiva o negativa la expresión del NA, que es un factor de transcripción. Este factor de transcripción se trasloca al núcleo de la célula, en donde forma heterodímeros o dímeros, homodímeros, perdón. Aquí lo puse nada más como HOD haciendo referencia a homodímeros o HED haciendo referencia a heterodímeros porque son bastantes las proteínas, los factores de transcripción con los que NAC puede asociarse para comenzar ese trabajo de transcripción y comienza precisamente la transcripción de algunas enzimas antioxidantes como la superoxidismutasa y la catalasa. ¿Y esto con qué objetivo lo hace la planta? Bueno, pues con el objetivo de inhibir precisamente el estrés oxidativo, esas especies reactivas de oxígeno. De manera
Disculpa,
general,
doctora Herrera, estamos sobre el tiempo ya. Si puede terminar la presentación, gracias.
sí, ya vamos a terminar. Bien, entonces estas enzimas antioxidantes lo que hacen es que de alguna manera permiten la transformación de estas especies reactivas de oxígeno, por ejemplo, el superóxido mutasa, convierte los radicales superóxido a peróxido de hidrógeno y la catalasa los convierte a agua. Y de esta manera es como que la célula lidia con el estrés oxidativo. Y bueno, pues como lo menciono, se ha reportado que las auxinas están ampliamente relacionadas con estos procesos, aunque se desconoce todavía el mecanismo exactamente muy ligado de una cosa con el metabolismo pues, de las especies reactivas de oxígeno y esta relación que existe entre las ROS y las auxinas. Bien, les dejo mis datos y les agradezco mucho la atención.
Muchas gracias. Thank you very much. We're going to keep moving with our schedule and our speakers. I want to remind everybody that if you join a little late, we're going to be taking questions at the end of the first session. Please write them on the chat. We're going to keep a tab of them. We may be able to answer them as we go through, and we'll certainly go through them at the end of this session. Our next speaker is Dr. Wayne Hunter. He is from the United States Department of Agriculture, Agricultural Research Service, and he will be talking to us about emerging biopesticides, RNA interference-based strategies for insect pest control. And with that, Dr. Hunter, I'll give you the floor. Wayne, if you're speaking, you're on mute.
All right, let me try it again here.
Yep.
Look like it's working right. It worked before. You see the first slide?
Not yet.
Huh. Wait. I get a weird shit.
Okay, it's looking like it's coming in. Yep. And now if you can, we're seeing the presenter view. So if you can switch it
Again?
at the top on display settings and then swap.
That it?
Yep, perfect.
Okay,
Floor
sorry.
is yours. Thank you.
All right. I'm Wayne Hunter, a researcher with USDA. Been working in RNAi for over 20 years. I'm going to cover just some data on several different projects that were successful. I think we're all pretty excited with this. In 2023, we saw the first approval of an exogenous spray, double-strand RNA. So I know from my program, this is really good that the RNAi product door is open now. And for those of us that have been working with developing biopesticides for many years, we'd like to see these be fast-tracked out through the regulatory process. So at least it's starting to look promising. As most of you have probably done in the past, I started with genomes of insects, plants, and bacteria, and you pretty much had to do all the data mining yourself, all the design molecules yourself, based on past literature searches of what was critical to the organism that you're studying. I think with the emergence of RNAi and AI software this year, we're going to see the next 3 years accelerate this field of research tremendously. It's going to more than most of us can even imagine. And so that's also very exciting. As I said, I'm going to talk about some successful projects I've done and the molecules. Now, this is a short list of the molecules I work with. I primarily started with delivery to validate I could get these molecules into plants and then into insects. But I also work on stability and activity. So I work with incorporating modified nucleotides, and I follow the medical profession research quite heavily. If anyone's interested in seeing a review of the psyllid work, though, this review article we just published has most of the information on the hemipteran work. Okay, so with delivery, the problems in the past have been numerous, but with the new breakthroughs in RNAi aptamer designs, delivery has become less of an issue. So in 2008, I started a project with a company called Biologics. We had colony collapse disorder of bees identified in 2006 in Florida, and it was linked to a viral pathogens. Now, bees all around the world have 4 or more viral pathogens. The group I was working with had developed a therapeutic for Israeli acute paralysis virus. And so we screened this in the field. It was the first approved. It was conducted under the Food and Drug Administration because honeybees are considered livestock animals. And so it was an interesting experiment in the field, multi-state trial. And we were able to show that when you feed the bees the double-strand RNA therapeutic, you could reduce the virus and increase the number of bees which then resulted in more honey production. So it was a very successful project. That technology has since been bought by Crop Bear Science and then licensed to Greenlight Biosciences, which has just released the first sprayable. So it's gone full circle, and I'm excited to see it's out there with a commercial company. And then we worked with Embrapa. It was a LabX USA program where they team US scientists with Brazilian scientists. I hosted Dr. Andrade in my lab, who's a plant pathologist, and I'm an entomologist. So we brought different perspectives on how to develop the double-strand RNA. But within 3 years, we had designed, screened, and patented the first double-strand RNA for psyllid management. And so that was real exciting, and it was a great collaboration. I really marvel at the skills of the Brazilian researchers, and I love working with people from other countries because it brings a new perspective. Then I was put in charge as the science advisor for RNAi development for the whitefly cassava food security program in East Africa, which was primarily Tanzania and Uganda. And I flew over there and helped train students in RNAi bioassays at the Institute of Tropical Agriculture, IITA, in Dr. James Legg's lab, and worked with Dr. Lena Tripathi, who's now the director of Eastern African Hub for Biotechnology. We successfully did the genome of the whitefly, and we also patented 10 double-strand RNAs suitable for the management of whiteflies. Then I was approached, when I was doing the bee project, I was approached by a medical company called AUM Life Tech. Now, they wanted to branch out. They're doing antisense oligo management of HIV and Duchenne's muscle wasting disease with these short 22 to 24 nucleotide modified antisense oligos. And their modifications make it so that these will enter into cells without the need of any carriers or other bound agents, which is really, really a good trait to have. And they're also fairly stable and resist degradation. Now they're using an RNAi cleavage. That's what the trigger pathway, but it works really well. And you're able to target bacteria, viruses, anything with DNA or RNA, you can target with these antisense oligos, which was really beneficial because I was working with Xylella in grapevines and Liberobacter bacteria in citrus. And then we also could use these to target the endosymbionts in the insects. And we were able to have multiple patents across multiple insect and bacterial species. It was a very successful program. Following that, I had a grant where I had a technician, Salvador Lopez. We data mined out genes out of the mice data. The mice data from 1960, 1965 has a lot of really good information on critical genes for development. And we selected a gene that we data mined out of the psyllid genome data set. And we made an antisense oligo as well as a double-strand RNA and a modified double-strand RNA. And this core sequence of the microRNA was also 100% similar to the Spodoptera fall armyworm. And in all cases, when we feed it, we get excellent suppression of the pest. And of course, this was submitted for patent. The last project we had, I read a paper in 2013, and since I needed something to kill bacteria, and I primarily had been working with insect-transmitted viruses, I got all excited when I read this paper out of Sid Altman's lab at the University of Yale. And he's a Nobel laureate for describing ribozymes, which I tried playing around with ribozymes in 2010. But I called him up and I said, I want to fly up to your lab and I'm going to meet with you and find out more about these molecules. And so we set up an agreement, we got a grant, and we were able to show that with these molecules, which are more expensive to produce, and they do need a carrier because they're basically negatively charged, or they have no charge, and you have to give them a charge or they won't enter into cells. So it's a little trickier to play around with these. There have been some entomologists lately, 2014, 2018, 2021, that have started using morpholinos. Morpholinos are primarily used for functional genomic studies, but they do have a role in the management of pests and pathogens because they're so stable. So you'll see a lot of review articles that will say production is still a problem, but production is no longer a problem. I'm here to say, let's just put that to rest and not mention that anymore in review papers. Production issues have been solved. They can produce double-stranded RNAs in a wide range of ways, but when you do it synthetically, when I started, it cost me $5,000 to treat one tree. And now it costs me 30 cents to treat that same tree. So production is competitive with chemical insecticides, herbicides, fungicides. So it's doable. I have a cost breakdown of at least my track of work in that review article that I mentioned earlier. So when it comes to production, as I said, you can do it synthetically. And what I've done is focus on root delivery because in the field of study, nobody was working on root absorption. So I was adding it to the soil. But you can do it with GMO plants, which is the traditional way. But now there's people using bacterial expression systems and yeast and mosquitoes. Dr. Duchenne, Molly Duchenne's work, which has had excellent success in developing product. And like I said, there's enough. If you use a modified nucleotide, there's 100 natural modified nucleotides, so they're not GMO or anything. They're just natural modifications. Many of them can still be researched. No one's used them or tried them. And so that's excellent studies for students to move this forward. Of course, in citrus, they wanted sprayable. Like I said, I was doing soil applications, but they also wanted foliar. And a lot of people have worked on that. But what I needed to show in 2008 was that I could put this in the ground and it wouldn't automatically degrade. Everyone said you dump double-stranded in the ground, it will degrade. And we were able to show that you put it in the soil, in potted plants with soil or without soil, we could absorb it into the plants. It loads up into the xylem where there's very little RNase activity, so it remains resident and it would slowly leak out or be transported into the other parts of the plant where you could still detect full-length double-stranded up to 57 days. I mean, it was eye-opening for everybody at the time. So then, of course, like you heard in the previous presentation, you have to take it to the field to see if you're going to get a totally different thing in the field. The field has so much more. There's more organisms in the soil, and you have the weather and all kinds of things. So I poured double-stranded on the ground in about a 30-centimeter circle at the drip line so you could get the roots. But in Florida, the problem was it's so sandy, the water doesn't permeate down to the soil. So then you had to follow your treatment with 2 liters of water, just enough to drive it down to get it into the roots. But when you do that, you don't know how much is in the tree. And so then we started doing these trunk injections. Now, trunk injection, there's 2 companies that currently make a more automated system for this that are actually being tested. One of them's being tested in Florida in trees, and they're just using compounds that are antibacterial that are already in the commercial market, so they don't have to be approved. And they have identified like 80 compounds that work really well to kill Liberibacter. And so this is the will be the 3rd year coming up trial to look at yield data. For me, when I was doing the soil treatment, though, I could get 2 months. I needed to get 4 months is what we calculated to get suitable solid management. And so I started dealing with soil absorbents, any kind of absorbent I could add into the soil that would release the double-stranded RNA. I found that natural clays from this company, Oil Dry, which works in industrial absorbents, cat litter, and animal feeds, this is an excellent product. You can absorb your double-stranded RNA, put it in the soil, put it in around your plant. Every time it's watered, it releases double-stranded RNA. And the beauty of that is I went from 2 months to over a year of delivering full-length double-stranded RNA. It stays inside that pellet. It's protected from bacteria. We've even played it out in a plate in the sun for 3 days. You had no degradation because it's inside the pellet. UV doesn't degrade it. It's an excellent product. Not that I'm pushing it. I'm just saying this is the first report where we've been able to see double-stranded delivery beyond 35 days. This goes well beyond 60 days and over to a year and a half. This is going to be how it's going to have to be applied to really be cost effective because you don't have to drive over your field again and again and again, which costs the grower money every time he has to drive over the field. And finally, let me just end with this. So we were developing, we always develop like a phenotype trigger to make sure we treat the plant, we see the trigger, we can calculate time of delivery, time of induction of or disruption of expression, cytokine desaturase in plants, eye pigments in insects. And here you see the first CRISPR knockout where we did vermilion and you get a whitish section in the eye. And I had a postdoc I just hired who was into optogenetics, which had won the Nobel Prize that year. And he's like, why don't we, if it's changing the pigments, it's changing their perception of the world. And so we didn't get to pursue this very far. It's very preliminary right now. But we were curious if we couldn't use it in a push-pull strategy where you could push them off your crop and onto a trap crop. And so we data mined out all the options. We designed our double-strand RNAs. He had previously in his PhD conducted a trial on this insect phototube, and we used it for our RNAi disruption. And he did see significant differences in the movement of insects for some of the RNAi treated insects, specifically in the UV opsin, which makes sense. UV was going to have the highest impact on the biorhythms, circadian rhythms of the insect, which also affects development and activity. And so we were really excited about this. And this is a great project for some student or lab to pick up and pursue it. This probably has very wide application. Even if you adjusted it from visual, it could be to like chemosensory. There's just so many things you could do with this. And then with that, I'd like to thank my current team. Dr. Larson just left with our downsizing, and he got a job at the University of Florida. Douglas here is an excellent in bioinformatics. He's looking for a PhD, molecular science. If anyone has one, send it to me. I'll connect the two of you. And I want to acknowledge my technician who's been with me through all of this, and thank you.
Right. Thank you so much, Dr. Hunter. That was pretty amazing. Lots of questions. I'm sure they're going to be for you. Okay, we're going to move on to our next speaker, Dr. Carolina Delgado. She comes from the National Institute for Forestry, Agricultural, and Livestock Research in Mexico. And she's going to be talking to us about the analysis of intestinal content, a tool for plant health. And I want to remind everybody again that you can write your questions in the chat. Don't be shy. The floor is yours.
Ok, bueno, pues primeramente muchas gracias por la invitación. Hola a todos. A continuación les estaré hablando sobre el análisis de contenido intestinal y del impacto que este tipo de análisis tiene en estudios de dispersión y prevalencia de patógenos obligados en campo, como es el caso de Candidatus liberibacter solanacearum. Avanza la presentación, perdón, es que no.
No, todavía está en la primera slide.
A ver, perdón. Va de nuevo. Ok, perdón. Este trabajo se realizó en colaboración con investigadores de la Universidad Autónoma Agraria de Antonio Narro y con investigadores del Departamento de Agricultura de los Estados Unidos. Bueno, pues como ya muchos sabemos, Candidatus liberibacter solanacearum pues es un patógeno obligado que tiene gran importancia económica a nivel mundial, que es transmitido por el Psyllido bactericera coquerelli. Este patógeno tiene varios haplotipos, de los cuales los haplotipos A, B, F y J afectan los cultivos de solanáceas. Y el nombre de la enfermedad que ocasiona cambia de acuerdo al cultivo que es atacado. Este insecto y el patógeno en conjunto pueden provocar pérdidas en los cultivos que van desde el 50 hasta el 100%, afecta el rendimiento, la calidad del fruto y pues obviamente incrementa los costos de producción, lo cual ocasiona que el cultivo entre en crisis y posteriormente se abandone. Una de las zonas productoras de papa más importantes a nivel nacional aquí en México es la zona de Galeana, Nuevo León, que en los últimos 20 años, como podemos observar en la gráfica, ha tenido una disminución de más del 58% en la superficie sembrada. ¿Por qué se da esto? Bueno, pues principalmente se da porque no hay un control efectivo para el patógeno. Cuando nos damos cuenta de que nuestras plantas están infectadas, pues es cuando ya empiezan a mostrar los síntomas de la enfermedad y para entonces pues ya es demasiado tarde. Entonces, ¿qué se hace? Pues se dirige el control hacia el vector. Pero aquí hay dos problemas. El primero es que la dificultad para predecir cuándo y de dónde van a estar arribando los cílidos y que no tenemos la certeza de si estos cílidos ya están infectados con el patógeno cuando arriban al cultivo. ¿Okay? A esto se le suma pues que Bactericera coquerelli, el vector, pues tiene hospederos silvestres, que son especies de solanáceas y convolvuláceas mayormente, que sirven al insecto como hospedero, y pues algunos pueden albergar al patógeno. Cuando hablamos de hospederos silvestres, hay dos términos importantes para poder diferenciarlos. Burger y colaboradores son quien los proponen, y el primero es planta hospedera, que se refiere a todas aquellas plantas que le permiten al cílido poder desarrollarse desde huevo hasta su etapa adulta. Y luego están las plantas alimenticias, que son aquellas donde el cílido adulto se puede alimentar, pero no se puede reproducir ni desarrollar en ellas, por lo tanto, el tiempo que pasan sobre estas plantas es muy corto. Es importante tener en cuenta que las especies de plantas hospederas y las alimenticias pueden variar de acuerdo con la zona geográfica. Un ejemplo de esto es Solanum nigrum, mayormente conocida o comúnmente conocida como hierba mora. Esta planta en Nueva Zelanda han reportado que Bactericera coquerelli se posa sobre ella oviposita y las ninfas emergen, pero mueren en los primeros 2 estadios. Mientras que en Norteamérica esta planta está reportada como un hospedero ideal para el cílido, es decir, que le permite completar su ciclo de vida desde huevo hasta su etapa adulta. Algo muy importante en los hospederos es que su importancia va a depender de su abundancia, de qué tan cerca se encuentren a los campos de cultivo, de la capacidad que tengan estas plantas para albergar al patógeno y también de producir cílidos que ya estén infectados. Entonces, con base en todo esto, debido a que las pérdidas económicas que son causadas por Candidatus liberibacter solanacearum y por Bactericera, junto con la dificultad para controlarlos, hacen que sea necesario realizar investigación que esté enfocada a comprender los patrones alimenticios del insecto. ¿Por qué? Porque esto nos va a permitir conocer cuáles son las plantas de las que se está alimentando el insecto, especialmente sus hospederos silvestres, y esto nos va a permitir esclarecer cuál es el papel que juegan en la dispersión y prevalencia de Candidatus liberibacter solanacearum en campo. Y a su vez, pues nos va a permitir diseñar estrategias de manejo que sean más efectivas. Para esto, pues establecimos 2 objetivos. El primero fue inferir sobre las fuentes de cílidos infectados con el patógeno a partir del análisis molecular del contenido intestinal de los adultos. El segundo fue ya identificar en campo los hospederos silvestres del cílido y determinar si estos podían albergar al patógeno. Para esto, para el primer objetivo, lo que hicimos fue en noviembre de 2019 colectamos adultos de Bactericera en una parcela experimental de Physalis isocarpa. Estos cílidos se enviaron a Luzda, donde primeramente se analizó si estaban o no infectados con el patógeno. Se determinó el haplotipo tanto del insecto como del patógeno y posteriormente se analizó el contenido intestinal de los cílidos para conocer su dieta, es decir, saber de qué planta se había estado alimentando. Las plantas que arrojó el análisis y que no están reportadas como hospederos de bactericera se consideraron como plantas alimenticias. Por cuestiones de tiempo, no ahondaré más en las cuestiones de la metodología del análisis, pero esta información se encuentra publicado, ahí pueden encontrar más detallada la técnica, ¿ok? Aquí abajo les dejo la referencia. Bien, ¿qué fue lo que arrojó el análisis? Primeramente podemos ver que los insectos analizados los colocó en 3 grupos, ¿ok? En total se analizaron 21 cílidos. De estos cílidos, 20 se identificaron como haplotipo central y que contenían el haplotipo A de la bacteria, y que solamente uno era del haplotipo occidental y tenía el haplotipo B de la bacteria. El 57.14% de los cílidos analizados fueron positivos al patógeno, mientras que el 28.57% fue negativo. Se obtuvo un 14.28% de insectos con una lectura débil positiva a candidatos, por lo que muy posiblemente pues estaban infectados también. Como podemos observar, el 100% de los insectos se había alimentado de Physalis, lo cual es lógico porque recordemos que los habíamos colectado de una parcela experimental de Physalis isocarpa. También dice que el 100% de los insectos se habían alimentado de una especie muy similar a Solanum dulcamara, y digo muy similar o cercanamente a esta especie porque en la zona no tenemos presencia de esta planta. El 71.43% de los insectos se habían alimentado de plantas de otras 12 familias, ¿ok? Con esto, bueno, confirmamos que los cílidos se mueven entre hábitats cultivados y no cultivados, ¿ok? Que la infección del patógeno está asociada con la alimentación en una mayor diversidad de plantas, particularmente solanáceas. Esto quiere decir que entre más variada sea la dieta del insecto, más posibilidades hay de que éste sea positivo a Liberibacter solanacearum. Entonces, con base en esta información, nos dimos a la tarea ya de pasar a realizar estudios en campo. ¿Y en qué consistieron estos estudios? Primeramente, teníamos que identificar cuáles eran los hospederos silvestres de Bactericera coquerelli que se encontraban en campo. Para esto realizamos muestreos mensuales a partir de diciembre de 2020 hasta enero de 2023. En la zona productora de papa de Coahuila, Coahuila y Nuevo León, México, que es la zona marcada en verde que pueden ver en la figura. Ahí establecimos 9 puntos de muestreo y por punto de muestreo se colectaron de 5 a 10 plantas por especie. En el caso de los arbustos, por punto de muestreo se tomaron 15 arbustos y se tomaron 5 brotes por estrato de aproximadamente de 10 a 15 centímetros de largo, y pues todas las plantas se identificaron taxonómicamente. Posteriormente, pues teníamos que saber si estas plantas tenían la capacidad de albergar al patógeno. Entonces tomamos muestras de aproximadamente 2 centímetros de tallo raíz de las plantas. También tomamos hojas y algunos adultos que habían emergido de estos hospederos silvestres. Todas estas muestras se enviaron a Luzda, donde fueron analizadas. Ahora, ¿qué fue lo que encontramos? Confirmamos 2 hospederos silvestres, es decir, que anteriormente, o al menos en otras zonas, ya están reportados como hospederos de Bactericera, que son Lycium berlandieri y Solanum elegnipolium, y encontramos 2 nuevos hospederos que no están reportados para Bactericera coquerelli, que son Camaseraca coronopus, es un reporte a nivel género, y Physalis virginiana a nivel especie. Estos 4 hospederos los separamos en 2 grupos, en hospederos perennes caducifolios, que están presentes en campo durante la primavera, verano y hasta casi finales del otoño, y en hospedero perenne, que es Lycium berlandieri, que se encuentra presente en campo durante todo el año, incluido el invierno. Ahora bien, Con respecto a la presencia del insecto sobre estos hospederos. Si observamos la gráfica, podemos ver que el número de huevos incrementa sobre los hospederos silvestres en los meses de otoño-invierno, cuando no hay cultivos presentes en campo, y disminuye cuando hay cultivos presentes en campo, que son las fechas marcadas en verde, fechas donde hay cultivos presentes en campo. Y esta misma tendencia se repite con las ninfas. Incrementa en los hospederos silvestres cuando no hay cultivos y disminuye cuando hay cultivos en campo. Y tanto en la gráfica anterior, en esta y en esta, podemos observar que que los hospederos que presentaron un mayor número de individuos son Lycium berlandieri y Physalis virginiana, lo que nos indica que este insecto tiene cierta preferencia por estos hospederos. Entonces, como un resumen de lo anterior, podemos decir que Bactericera coquerelli sobrevive el invierno en Lycium berlandieri, posteriormente migra a los hospederos perennes caducifolios, y al final de la primavera, cuando ya se establecen los cultivos de solanáceas, el insecto migra de estos hospederos silvestres hacia los cultivos y se mantiene ahí hasta que estos cultivos ya están listos para ser cosechados o son cosechados. Entonces el insecto se ve obligado a regresar a los hospederos perennes caducifolios, y cuando estos desaparecen del campo a finales del otoño, principios del invierno, pues el insecto se ve obligado a regresar a Lycium berlandieri y empieza a repetir el ciclo. Ahora, con respecto a que si estos hospederos tienen o no la capacidad de albergar al patógeno, bueno, pues encontramos que 3 de los 4 hospederos silvestres dieron positivo al patógeno, que son los que podemos observar aquí. Solanum eleanifolium fue el hospedero que presentó un mayor porcentaje de infección, pero aquí pasa algo curioso. Si recordamos las gráficas anteriores, pues es de los hospederos menos preferidos o menos buscados por el insecto. Y bueno, también encontramos que adultos de Bactericera coquerelli que emergían de estos hospederos silvestres eran positivos a Liberibacter solanacearum. Hay casos ya reportados donde plantas de Solanum eleanifolium, de Lycium berlandieri, Physalis virginiana, dan negativo al patógeno, pero cílidos que emergen de estas plantas que dieron negativo son positivos. Aparentemente se debe a que los niveles de la bacteria en la planta pues eran demasiado bajos, que no los detectó el análisis, pero eran lo suficientemente altos para que el insecto los adquiriera y pues se mantuviera infectado. Pues en conclusión, el análisis de contenido intestinal nos permite conocer cuáles son las plantas que sirven de alimento al insecto vector y así poder identificar las especies que sirven de reservorio al patógeno. Saber qué plantas son las que atraen al insecto vector nos permite implementar estrategias de control para reducir la presencia del insecto y su capacidad de transmisión. Pero es fundamental realizar también estudios de campo que nos permitan validar los resultados del análisis en condiciones reales, para así poder identificar los patrones de comportamiento del insecto y ajustar las estrategias del control según el contexto agroecológico. Y pues eso sería todo de mi parte.
Muchas gracias, doctora Delgado. Right, we're running a little bit behind on our schedule, but we still have 2 speakers, and we're going to be switching gears here to start discussing RNAi technologies on weed control. Our next speaker, Dr. Martin Laforest, he comes from Agriculture and Agri-Food Canada, and he's going to be introducing the concept and talking to us about RNAi and weed control. Okay, the floor is yours.
Thank you. Sorry. Good afternoon, everyone. You can hear me, I suppose. I hope so. Thank you for the opportunity to share my work with you today. As a weed scientist, my research primarily focuses on herbicide resistance. It's a growing challenge that threatens the effectiveness of herbicides. This issue led me to explore RNA interference, or RNAi, as a promising alternative for managing weeds. The research I'll be presenting today is the results of a collaboration with Gagan-Pri Dariwal, Marie-Josée Simard, and Charles Geddes. Of course, I will be talking about topical application of RNA. So farmers and producers must cope with extreme weather conditions, pests, and diseases. But the greatest threat to food production is weeds. Fortunately, they've been kept in check very efficiently with herbicides. This slide highlights the dual nature of herbicides in agriculture. Herbicides are essential tools of modern agriculture. They help protect crop yields by effectively controlling weeds. This allows for more intensive farming and boosts overall productivity. Herbicides are also vital for soil conservation practices, such as no-till farming, which helps prevent soil erosion and preserves soil health. However, there are significant downsides. The use of herbicides raises both environmental and health concerns due to the chemical residues and potential toxicity. They can also reduce biodiversity by impacting non-target plants and beneficial organisms in the ecosystem. And another major issue is the development of herbicide-resistant weeds, which makes these chemicals less effective over time. It leads to increased use of herbicides and also stronger products. This graph shows the number of unique herbicide resistance cases over time. It's been steadily increasing since the late 70s. It looks as if it's tapering off here in recent years, but it is not. It is just a delay in reporting cases. Till now, there are 537 herbicide resistance cases reported. Resistance is developing to several groups of herbicides. Here in blue, we have resistance to glyphosate, an herbicide for which resistance was thought impossible to evolve. The chance of developing resistance is a factor of how often the herbicide is used and onto what acreage. This graph shows in what year the different herbicide groups were introduced. Unfortunately, there was a very long period of time, almost 40 years, where no herbicide groups were introduced, from the early 80s to early 2020s. And recently, the group 28 was introduced. Because of the lack of new groups of herbicides, because of resistance, and to reduce our reliance on the chemical control, it would be beneficial to develop a new approach to weed control. Dr. Hunter already presented some examples of controlling insects with double-strand RNAs. An article published in 2015 showed us how it also could be done. This study shows that foliar application of double-strand RNA on targeting the Colorado potato beetle, CPB, the actin gene, causes rapid feeding cessation and high mortality in second and star larvae, with longer double-strand RNA fragments being more effective and 5 microgram per leaf achieving near complete control. In greenhouse trials, a single application protected plants for at least 28 days, and was not really readily washed off, and also tolerated some UV light. It did not move systematically, however, so full leaf coverage was required. Around the same time, BioDirect and Monsanto presented a weed control approach based on double-strand RNA. EPSPS is the name of the gene that codes for the protein that is targeted by glyphosate. Adding EPSPS targeted double-strand RNAs to glyphosate significantly improves control of glyphosate-resistant Palmer amaranth and water hemp in field trials, with treating plots having much less weed growth compared to those treated with herbicide alone or left untreated. This proof of concept demonstrates that double-strand RNA can be effective against both palmer amaranth and water hemp in field conditions. I could not find a commercial product or more commercial information about this technology, however. I will not explain this slide. It's post-transcriptional silencing. It was already covered by Steve in his previous presentation. But I will elaborate a little bit more because there are two more pathways showing how double-strand RNA can control gene expression. The pathway on the right is what Steve described. It involves DCL4 or dicer-like 4. In the middle pathway, dicer-like 3, DCL3 produces 24 nucleotides, small interfering RNAs that associate with argonaute 4 and target DNA. This leads to DNA methylation with the help of RNA polymerase 5, causing stable, heritable silencing at the DNA level. In the left pathway, a long double-strand RNA is processed by DCL2 into 22 nucleotide long siRNAs, which are loaded onto argonaute 1. This complex can recruit RDR6, an RNA-dependent RNA polymerase, to amplify the silencing signal, a process called transitivity, generating more small interference RNA targeting the same or nearby sequences. The more siRNA there is, the more likely they can move in the plant. Indeed, siRNA can move inside the plant. They are produced in source region, like mature leaves, and they can move through the phloem via the companion cells or sieve elements to distant tissues, the sink regions such as tips or the growing tips or young leaves. This systemic movement of siRNAs ensure that gene silencing is not limited to where it is an initial trigger occurred, but can spread throughout the plant, allowing both post-transcriptional and transcriptional gene silencing to take place in multiple tissues. Plasmodesmata allow the small RNA to move from cell to cell, promoting a wide-ranging efficiency in silencing. In short, transitivity amplifies systematically spread the silencing signal throughout the plant, ensuring robust and widespread gene regulation. Yeah. I'm presenting all of these mechanisms because we may need to tweak them, or many of them, to develop an efficient RNAi herbicide. There are significant challenges to developing an RNAi-based herbicides. We need to ensure that enough DNA is delivered into plant cells to achieve effective gene silencing. That may require large amounts of double-strand RNA. It is also crucial that to target gene whose expression will make the weeds less competitive with crops. Lastly, transitivity will likely play an important role to prevent untreated leaves from compensating for the silenced ones, which could otherwise reduce the effectiveness of the treat. The double-strand RNA must penetrate the cuticle, the cell wall, the cell membrane, but not enter the vacuole. As Dr. Hunter mentioned, delivery is key. To improve penetration and effectiveness of double-strand RNA treatments, we have explored several strategies. Physically, we have tried abrasion techniques, using materials like sand carborundum, sodium bicarbonate, walnut shell grit, and corn cob grit. Chemically, we've used various adjuvants and solvents, including the non-ionic surfactant Li700, transfection agent lipofectin or lipofectamine, and polybrine, a cationic polymer that enhances uptake in mammalian cells. Additionally, we've tested nanomaterials such as kaolin, which is a layered silicate clay, and employed also cell-penetrating peptides, CPPs, to facilitate double-strand RNA delivery. You can see some of the CPPs that we have used. They're on the screen right now. Although we have not tried bioclay, which is a layered double hydroxide, it's been shown to be effective in other applications. Selecting the appropriate gene for the development of an RNAi-based herbicide is very important. One option is to target genes that are already known herbicide sites of actions. There are some examples on screen: ALS, HPPD. Dr. Hunter talked about PDS, phytoandesaturases, a little bit earlier. We could also target genes that are essential to the weed. The sequence used must be different from the crop in which it is used, of course, to avoid off-target effects. Additionally, the optimal design for the RNAi trigger itself needs to be considered. For example, whether to use 21 base pair small RNAs or longer double-strand RNA fragments ranging, let's say, from 300 to 800 base pair. We can use also a cocktail of several different double-strand RNAs targeting multiple genes or targeting the multiple regions of the same gene. That may help improve the effectiveness and durability of the RNAi response. Dr. Hunter shared with us that the cost of double-strand RNA molecules have come down significantly. This is very good since we need a lot when considering the field scale. To improve the effectiveness and stability further, protective carriers like BioClay can shield double-strand RNA from degradation. Additionally, using double-strand RNA during design that will trigger transitivity may improve gene silencing. Here are some examples of the abrasion tests that we did. On the left, this is the surface of the leaf with air only, so it looks good. In the middle, it's a leaf that were treated with walnut shell grit. This could hurt too much the crop in field settings. There's a lot of damage. Sand gave us a nice shave over the surface of the leaf. Here is an example with common ragweed. We have tested 3 different formulations. On each of these pictures, the plant on the left is without double-strand RNA, and the plant on the right is with double-strand RNA. We can see in these 3 examples that there was a growth retardation when double-strand RNA was added. Unfortunately, this is not enough to control the weeds, as they have a great ability to recover from these treatments. Our colleagues in Alberta have also tried to control kochia with RNAi. They have some interesting phenotypes. The top row are the controls. So on the extreme left, we have a resistant kochia, resistance to glyphosate. And it was sprayed with glyphosate, and there's no symptoms. Glyphosate was sprayed on these resistant plants, treated also with cell penetrating peptide, and there are no symptoms. There was a EPSPS specific double-strand RNA put on these plants and treated with glyphosate. Again, no symptoms. They have used a positive control, so phytoandesaturases plus a cell penetrating peptide. This was not sprayed with glyphosate, but you can see here whitening of the leaves, and the younger leaves are bleached. We have 2 triggers for the EPSPS, EPSPS1 and EPSPS2. So if glyphosate-resistant kochia was treated with EPSPS1 plus a CPP and glyphosate, there was no symptoms. If the plants were treated with EPSPS2 or EPSPS1 plus EPSPS2 and CPP in both cases, both treated afterwards with glyphosate, bleaching occurred again. The young leaves were white bleached. So there seems to be some movement in the phloem, but there's no lateral movement of the dsRNA in these experiments. So we've been working to develop this promising technology to control weeds, but we're not there yet. There are many questions and challenges ahead of us. Once it is shown to work in the lab, we will need to bring it to the field. How will it scale? Is it safe to use? What are the other plant species? Will formulation be safe for farmers? And finally, there are regulatory issues, but these will be discussed later on today. On that, I'll stop here. Thank you for listening, and I'll be happy to take your questions.
Thank you so much, Dr. LaForest. We're going to move on with our last speaker. And I'll remind you again, we're going to have a few minutes for question and answers before the break. So please put them there and we'll go over those after this presentation. So our next presenter, Dr. Matthew Tankus, he comes from the United States Department of Agriculture, Agricultural Research Service, and he's going to be talking to us about genetic biocontrol tools for invasive pest plant management. And the floor is yours, Dr. Tankus.
Sounds good. Can you hear me okay? You see everything?
Yes, we can.
Awesome.
That's great.
Okay. All right. Well, thank you all. Thanks for the introduction, Yasmin, and thank you for the invitation to come speak to you today to tell you about some of the exciting research we're doing at the USDA Agricultural Research Service, specifically looking at novel genetic biocontrol tools for invasive plant management. So invasive weeds have a huge impact to our agricultural systems. They contaminate our harvests, they compete for limited resources, and they displace native and beneficial plant communities. And I included images of grazing livestock because rangelands are some of the most challenging environments to manage non-native invasive weeds due to the sheer size, the topography, and rugged terrain of these environments, and also the diversity of plant species that just makes it very challenging using conventional tools. But in addition to the agricultural sector, we also have a big impact across all the different facets of our life. So we have agriculture, but also infrastructure. So invasive weeds can really promote the intensity and frequency of wildfires with their abundant flammable vegetation and their ability to quickly reestablish following these disturbance events, which just cascades it into future years. Invasive weeds also play a direct role in human and animal health by harboring pests that vector animal and human diseases such as ticks. And then lastly, biosecurity risks, because when an invasive weed is introduced into North America, it's not just that given species that's introduced, it's all of those microorganisms that are co-introduced, whether they're bacteria, fungi, or viruses, whether they're new genotypes, new species, or even new pathotypes and pathivars that could potentially risk damage to our agricultural systems. So altogether, we really need some innovative biological solutions to control these invasive weeds that colonize these large, inaccessible, complex terrains, such as our rangelands, forests, or wetlands. And that's some of the work we're doing at the Foreign Disease Weed Science Research Unit. So we're located at Fort Detrick in Frederick, Maryland. And I'm the lead scientist with the Weed Biological Control Group. And our group primarily focuses on plant pathogens as host-specific biological control agents, but we also investigate a variety of other aspects of this research with conventional, molecular, and then today what I'm going to talk about is more of the technology-driven tools that we can use for weed management. And as today's talk is about RNAi and pest management, this is one of the things that we're trying to evaluate, similar to what Martin was just presenting on. And our goal is to hijack the plant's defense system and use it against itself. And we think it's going to be, we think a layered approach that incorporates RNAi suppression, microbes, and nanotechnology is going to be the most effective way to really attain weed suppression. And I want to highlight the images on the bottom because if we use RNAi technology to suppress, say, a given defense-related genes or whatever it may be, you might not see a strong phenotype on the plant, and the plant's still reproductive and will persist. And similarly, if you have a plant and you infect it with a weak pathogen or an opportunistic pathogen, again, you're not going to remove that plant. But it's that additive effect of suppressing its defenses and hitting it with either an endemic pathogen or a weak opportunistic pathogen that you can result in true plant death. So in order to see if this is actually going to be a viable tool, we wanted to see, can topical double-stranded RNA be a viable tool for this kind of approach? So we collaborated with Michelle Heck, another ARS scientist in Ithaca, New York. And she has a lot of these VIGs, or virus-induced gene silencing constructs, working with Nicotiana and other species. And we really wanted to focus on suppressing genes that have already been suppressed in a VIG system and that produce severe phenotypes, because we wanted to essentially fail fast, target the genes that are going to make the most severe phenotypes to kind of screen this process and how to optimize it to see if this is going to be species specific and an efficacious tool for weed suppression. So we looked at a variety of gene targets for these systems. And for the beginning, we wanted to utilize a diverse array of plants. So first, we wanted to use Nicotiana benthamiana kind of as our positive control, since these constructs were constructed and tested in Nicotiana with VIGs. It's a nice positive control to see, can we replicate that using topical applied double-stranded RNA. We also included Arabidopsis. It's a nice model system, and a brassica. And we included Arabidopsis because the target weed we wanted to demonstrate this on was garlic mustard, another non-native invasive brassicae plant. So, but one of the big things that's really different about the plants, and Martin was discussing this, was just the diversity of plants out there and the plasticity of the genomes. Some of these are very complex polyploids, and a lot of the targets, whether they're redundant, whether they have other functions, whether you're going to get compensation from other leaves that are not showing or expressing that phenotype as much, and also how is the RNA going to be modified in the given environments you're wanting to apply this on. But we noticed that leaf structure variation and composition also played a huge role, with some of the plants having a thick cuticle layer really, or hydrophobic aspects really limited the application. But our selected plants, especially garlic mustard, have a very tender leaf. It's not waxy, and it can readily uptake the material applied. But one of the things and challenges we did see is that the influence of plant and leaf age tends to play a big role in this. And is that related to the tissue and age-specific RNA expression levels and the environments that they're in? And ultimately, this really resulted in inconsistent phenotypes. And that was a challenge we were trying to always overcome, is sometimes it would work well in a given plant, or you would see it on some leaves, but not the other leaves. And then how long is that going to persist with the non-affected leaves supporting those affected ones? And here's just a quick little image of this. So on the left, we have, this is garlic mustard. We have our empty vector control sprayed to the leaf of garlic mustard, and we don't see any phenotype. And then on the right, we have our double-stranded RNA that is applied, and it's targeting the PDS gene, so for photobleaching. And if we look at the top macroscopic aspect of the leaf, we see some symptoms. It's not as severe as what we were anticipating, but we see the symptoms, we see the impact. But in these cases, we would see more of the younger leaves showing that suppression of the gene, similar to what Martin was explaining as well, with the newer leaves, not always the older leaves. And going to other conferences and talking about some of this, I actually connected with a variety of other scientists who are also interested in looking at RNAi as a weed management tool. And a variety of scientists from the United States and then also Australia. So we wanted to get together and actually have a workshop to discuss the challenges and what works with RNAi of invasive weeds. So we actually met up in Australia last year in Brisbane, and we had our workshop together, going over the research and seeing what works and what doesn't. And one of the big things or big takeaways that we took was that everyone's appearing to have that same challenge of inconsistent phenotypes. And even though we see gene expression reduction, that doesn't always translate into a phenotype. So it really complicates this and makes it questionable as if we apply topical double-stranded RNA, are we going to get enough of a phenotypic response to really impact plant fitness in a field environment? And that's the million-dollar question, and how viable is that going to be as a tool? And I wanted to bring up this last image, which is looking at the VIG system, or virus-induced gene silencing. So this is a common tool used in plant molecular biology to suppress a given gene instead of knocking it out. But we're using RNAi, but you're using a virus that's going to persistently express it. It's at high titers, and it also can move systemically. But even in these very optimized VIG systems, you see this variability of phenotype. So in this image, this is of soybean from a colleague doing work with VIG's constructs, again, targeting the PDS gene as a positive control. And you can just see the variability of phenotypes, not even within a leaf, but within between leaves, within plants. So again, even in these very conserved, optimized VIG systems, phenotypic plasticity and variation is common. So it really makes it a challenge to see this as developing as a very effective weed management tool. So we're kind of shifting gears and focusing resources into different projects. And instead of playing with expression levels, we were wondering, well, why can't we just knock out a gene? Not play with suppression of the gene, but truly knock it out. And you might be wondering, well, how are you going to do that as a weed management tool? So what we're thinking of is actually investigating a weed suppressive gene drive as a new weed management tool. So this is a little different from the RNAi, but it kind of segues into that because of trying to use a different approach to have weed suppression. Now, the challenge with a knockout and putting it into a population is if your modified gene is introduced, your offspring only has a 50% chance of inheritance. But with the CRISPR-based gene drive, we can essentially drive that modified gene into the population and theoretically achieve 100% inheritance. And again, similar to RNAi, this can be species-specific, and you're going to reduce the likelihood of collateral damage to non-target species. But in this case, we would knock out a gene and not just suppress it. So that's some of the current research that we're investigating is, can we develop a safe and efficacious weed suppressive gene drive? So we're actually looking at the invasive weed common tansy, which we're looking at as kind of our model system. And there's some other plants that we're also in the pipeline that we're investigating, but this is our initial model system that we want to evaluate because it has a lot of biological qualities to it, such as being sexually reproducing, self-incompatible, diploid, prolific seed producer, and a short life cycle. It also has a lot of agricultural impact, where it's a noxious weed, but it's also toxic to livestock, impacts milk quality, induces abortions, and also is a reservoir for chrysanthemum viruses, which is a big issue. And in the bottom schematic, so our ultimate goal is to develop a gene drive modified organism that would be female sterile, so it would not be able to make viable offspring, but still male fertile. So that viable pollen is able to pollinate and spread the gene drive into the population. So any subsequent offspring are going to be female sterile, but again, that pollen is going to move. And after subsequent generations, you could essentially eradicate the species or this invasive weed in the environment by making them entirely female sterile. So the first step in developing a gene drive is, can we even transform this invasive weed? And with a lot of these invasive weeds, we don't know much about them. We don't have a lot of that background molecular information. So with tansy, we had to actually sequence the genome, do RNA-seq analysis, annotate the genome. And then with collaborators at the University of Minnesota, we needed to see if we could even grow tansy in tissue culture. Because in a nutshell, we need to have an individual plant cell, introduce our foreign DNA into it, and then make that individual cell grow into a fully transgenic plant. And you can do this through tissue culture manipulation by controlling the hormones and the different stages of development. Long story short, we were able to successfully grow tansy in tissue culture, insert foreign DNA into it, and get transgenic plants. And now recently, I have a postdoc who's actually doing CRISPR-Cas gene editing. So now we're targeting with our genome, we're actually targeting several female-specific fertility genes to knock out and to characterize, because we need to ensure that we're not going to impact pollen production, but we'll make them sterile, female sterile, but then also have no other fitness costs because you want this to be viable in the field. And this is kind of the approach that my postdoc has been taking with agrobacterium infection, transform cell selection, regeneration, and then shoot elongation and root development. And we currently have several individual transgenic constructs that we're currently characterizing and validating. So hopefully you see all of the, whether it's RNAi or a gene drive, all of that research that goes into investigating and developing a new novel tool. And this is just some of the work that goes into the gene drive was tissue culture optimization, transformation, sequencing the genome, CRISPR gene editing, and then also looking at phenotype sequence variation, looking at a population sequencing studies. So we hope to actually narrow this down. We're characterizing, hopefully have this in a gene drive modified organism and be able to evaluate this in our biosafety level 3 high containment research facility to see how safe and efficacious this tool can be to ultimately control weeds in these challenging environments. And with that, I just want to thank ARS and APHIS for funding support, University of Minnesota and Michigan State University collaborators, and Demiro, Jamie, and Michael for all their work in these projects. So thank you.
Great. Thank you so much, Dr. Tankos. And with this, we end the presentations for the first session of this workshop. We're going to go over questions now. So I'm going to start with the first one. This one comes from Alma Tovar Diaz. Good morning, Dr. Robinson or Hunter. What materials to date are useful to avoid the degradation of double-stranded RNA in agricultural environments? The question is for Dr. Robinson or Dr. Hunter. So you can unmute yourself and help us out here. Yeah, go ahead, Dr. Hunter.
Yeah, I sent an email out. So there's been, okay, so the studies in RNA have been going on since 1960. And so when you start reading the reviews that start around 2005 with the antisense oligos for human medicine, that's the literature I primarily have been following up till today, there has just been numerous medical advances. And so I gave a list there there's a kit you can buy for those of you who want to improve the double-stranded RNA you already have. You can use that kit, and what it does is it prevents the first-tier enzymes that would degrade double-stranded RNA, the enzyme A level. It stops those enzymes, and I find that you will get a 15 to 25% increase in efficacy of your RNAi. And what it does is it adds a modification in the cytosine and uracil at the 2 position with a fluorine. Nature loves fluorines up to a point, then they become toxic. But fluorines, cells will absorb fluorine. They like fluorine. Biochemists will tell you, if you want to get something in a cell, add fluorine. If you want to get something in bacteria, you add iron. And so with these 2 strategies, you can target, a multiple of things. And then there's the shape. We didn't even get into shape. Shape's a whole another conversation. But that's the easiest way to show to yourself and to improve your RNAi is to get that kit, get it in your hands, make your double-stranded RNA, and prove to yourself that it improved it. And then there's other modifications, but that would be my advice to do your first step, just to show that you can do it. When you improve it, that new molecule is also patentable, right? So every time you do an improvement in your molecule, keep track of what you're doing. Regulatory people do not like you to hook something to your double-stranded RNA. I work with a company where we try to get our molecule, it's a small 7 amino acid peptide, first classified as an inert molecule. They could be used in food. So then we would not have to worry about it when we went for a biopesticide. But anytime you take something and attach it to something else, regulatory is going to hit you with an extra layer of regulatory requirements. So anyway, that's my answer.
Right. Perfect. Thank you. And it's great because we're going to be addressing those regulatory questions in the next session after a short break. We have a question from Xinyi Marzano. I was wondering, what is the mechanism behind new leaves being resistant? And I believe this is for the last two presenters on weeds.
If I may start, I think the older leaf will have a thicker cuticle. Maybe just the structure of the leaves will be less prone to let the double-strand RNA penetrate. So younger leaves will react better to a treatment, and also they are more active transcriptionally. So I think they react more because of that.
Okay, thank you. I do have a question for probably both of you as well. And I was thinking about, the changes in phenotypes for weeds. But what if you hit it with a lot of a larger dose or multiple doses, or does that do anything to make that phenotype changes a lot stronger to control the weeds?
Yeah, killing a weed is difficult because it's killing a plant in a plant environment. You need to make sure that the crop is left untouched. So I think if you put too much double-strand RNA, the molecule itself will become toxic to whatever at a certain point. I think you just can put so much on it, but that's my view.
Yeah, one of our recent experiments, that was our question, was like, maybe there's just not enough on it. So we put milligram levels per plant subsequent days in a row.
Mm-hmm.
And essentially, yeah, you see a response to just that, even with the empty vector control, double-stranded RNA. You do see a minor response. So I agree with Martin. It's challenging to get that balance to see enough of an impact without just overwhelming the plant just entirely on its own.
Okay, I see. All right, and then I'm going to take one more question, and we're going to then take a break for 5 minutes. And I apologize for cutting your break a little short, and then we'll jump into the regulatory aspects of RNAi technology. So this question comes from Justin Pahara for Dr. Hunter. You mentioned recent design principles for RNA that support delivery. Can you expand on these? And I know you might have already answered some of these in the chat, but if you can just give us a few comments there, that'll be great.
Right. So that kit I mentioned that I sent out again, and it's in your chat list, and anyone can email me and we can set up a time to talk, because if I know better what you're trying to do in your system, I can address many of your problems or concerns. But again, so to get it into the, if you're talking about a plant, or in my work, I have found that soil applied double-strand RNA will enter the plant and give me 2 or 3 times more concentration than a foliar applied. When you do foliar applied, we screened like 85 different surfactants that are used by the industry. Most of them degrade double-strand RNA. So there's only like 4 that really work. And same in tank mixes. You can't give double-strand RNA product to a grower that we tried to run it through an irrigation line if they're also going to add other stuff in there. You always want to see what's the system that you're trying to introduce your treatment into, so you know what the grower is doing. You don't want to spend 5 years developing something only to have the grower add a chemical that destroys the product. And so know what system you're getting into, first of all. Second of all, like I said, do that kit. That will extend your persistence. Persistence has been the #1 thing we've been working on, trying to get molecules that persist. you can now incorporate. So if you incorporate a 2O methyl, a 2O-ME, that's not out, nobody has a patent on that. Anybody can use it. It's a natural modification that we did on a microRNA, and you just use it on, like, you have 24 nucleotides or 27 if you're doing a pre-mRNA. You only need to hook it on about 4 nucleotides on each end, so it's very cost effective. But you could probably even reduce that down to 3. And so those modified nucleotides prevent degradation. So if I'm treating a plant or feeding an insect 10 nanograms, a lot of that's, if you treat a plant, it goes everywhere. So if your insect's a phloem feeder, a lot of that product is not where the insect's feeding. So I calculated in psyllids, they were only ingesting 2 picograms a day feeding, because my molecule goes systemic all over the plant. And so it needed 4 days to get to 8 picograms inside the insect before it would induce a strong enough RNAi that we would see an effect on day 8. And so, these are the kinds of things you have to think about. If you're doing short molecules, you can do antisense oligos, you can do a pretrial. I do in-lab testing. You order it online, it's about $500-600, because we're always working towards patents. Then you test it yourself in your system. And then there's, like I say, there's companies. AUM will work with you to do modifications. There's other companies that will work with you to do modifications. If you don't have a department that can patent stuff, then you work with a company like AUM and let them shoulder the cost of patent. If you work with ARS, we shoulder the cost of the patent. There's all these options that, of course, I don't know, current environment, I'm not sure, but in the past, were very helpful at getting your research from the lab to the field. That's what ARS does. We want you to get your technology from the lab to the field. Anyway, we're getting off from the activity and persistence, but if you use modifieds, you're going to increase it automatically.
Perfect. Thank you so much. Yeah, and I think there might be some commercialization questions after the next session. Definitely, there's some already. So thank you to all the speakers for the first session. It was a great session, great range of topics on RNAi and other genetic control for plant pests, right? Weeds, pathogens, insects. What we're going to do here is we're going to take a very short break, 5-minute break, and then we're going to come back, I would say at 2:09 Eastern Time to continue with the second session where we have 3 speakers that are going to discuss regulatory issues. So with that, we'll do a 5-minute break and we'll come back in 2:09. Thank you.
On the regulatory challenges and aspects and processes, where we have 3 speakers from US, Canada, and Mexico. So over to you, Jaime.
Yeah. Okay. Good afternoon, everybody. Thank you for your patience. But we are pretty much in the second part of this workshop. It will be only 3 more speakers, so hopefully in less than an hour we'll be done. So, but I'm really glad that we have already heard some of the previous speakers, and it was clear that developing an RNAi technology is not easy. But at the same time, I'm happy to present that the next 3 speakers will be talking about regulation, And so kind of both fields are developing in a similar way, which is good, because by the time a good RNAi technology is available, it should be already a regulation in place. So with that in mind, I would like to introduce the first speaker, is Dr. Nina Ortiz. She is from the Environmental Protection Agency in the United States, and her topic is about EPA regulatory considerations for topical RNA applications. So, Dr. Nina, the audience is yours.
Great. Thank you so much, Dr. Covarrubias. Hello, everyone, and good afternoon. My name is Nina Ortiz, and I'm a biologist in the Emerging Technologies branch at EPA, where I conduct human health risk assessments for pesticide products developed using novel biotechnology. Today, I'll be discussing EPA's regulatory considerations for topical RNA applications. Next slide, please. For a bit of background here, the regulation of pesticides by the EPA falls under two overarching statutory authorities, FIFRA and FFDCA. FIFRA, which is the Federal Insecticide, Fungicide, and Rodenticide Act, is the licensing statute. Under this law, EPA evaluates pesticides for their potential effects on the environment and human health, and also regulates their development, sale, distribution, and use. Occupational, bystander, and residential risks are also evaluated under FIFRA. FIFRA is unique in that it is a risk-benefit statute. That means that some risk may be tolerated if there are other benefits that may outweigh this risk. The second statute is the Federal Food, Drug, and Cosmetic Act, or FFDCA, and this is where EPA evaluates pesticides which are proposed for use in food or feed. In conducting assessments under FFDCA, EPA considers all anticipated dietary exposures, residential use, and other outdoor uses. Under FFDCA, EPA evaluates food safety and can establish the maximum amount of pesticide chemical residue that is safe for human consumption. Next slide, please. In order to make the safety findings for FIFRA and FFDCA, EPA's risk assessment paradigm for human health and the environment follows the equation risk equals hazard times exposure. Part of that review includes product characterization, which forms the foundation for both the human health and ecological risk assessment. This establishes the identity of the end-use product, the origin of the trait, the mode of action, and also the manufacturing process of the product. When looking at human health risk, the hazard side of the equation will look at components which would inform on any health effects the product may have. This includes looking at dose response and acute toxicity studies, and where applicable, taking into consideration the existence of a history of safe exposure for that particular active ingredient. The exposure side of the equation considers how much, how often, and the ways in which exposure occurs. This includes evaluating the type of product. Is it aerosolized, or is this something like a plant-incorporated protectant, where the active ingredient is expressed within the plant? Looking at ecological risk, the hazard and exposure components resemble those for human health, such as high-dose testing and a history of safe exposure, with considerations specific for non-target organisms, such as soil degradation studies and habitats for non-target organisms. When looking at the risk equation as a whole, if either the hazard or the exposure is low, then the overall risk is low. The framework of this risk assessment paradigm can also be used as a foundation when approaching the risk assessment of novel pesticides. Next slide, please. Over the past 30-ish years, EPA has seen a variety of biopesticidal products developed through biotechnology. And some examples include genetically engineered microbes and insects, topically applied proteins, topically applied RNA, and plant-incorporated protectants, also called PIPs, where the pesticidal substance is produced in the plant. Historically, the vast majority of the pesticidal products that our branch sees have been PIPs, some of which utilize RNA-based mechanisms for disease resistance, such as honey sweet plum, which has been engineered to be resistant to plumpox virus, and MON 87411 corn, which is protected against feeding damage caused by corn rootworm through expression of a double-stranded RNA called DV-SNP7. Next slide, please. Since the registration of DV-SNP7 in 2015, EPA has gained a lot of experience in assessing risk for double-stranded RNA produced in plants. For dsRNA pits, we know that the RNA is naked, so to speak, meaning that the RNA molecule itself has not been chemically modified. In fact, these dsRNAs are produced in the plant and are applied as part of the plant. So because of these properties, the potential routes of exposure are tied to the biology of that plant, and a history of safe exposure can be demonstrated since ultimately this RNA is no different than RNA molecules, which are a normal part of human and mammalian diets through the consumption of RNA that is naturally present in food. More recently, EPA is seeing an increased number of pesticidal products utilizing topically applied RNA. Unlike the PIPs I mentioned before, these types of RNA molecules may vary in their identity, manufacture, formulation, and application, depending on how the developer is envisioning their product. So while these pesticidal products are also RNA, because they are applied differently and they may not be equivalent to the RNA found in food, there may be some variables that are different from the RNA PIPs that have been previously evaluated. And so this must be taken into consideration by the developers of these products when assessing the potential for human health and ecological effects of their product. Next slide, please. To assess the hazard portion of the risk equation for human health, the potential for sequence-specific effects of the RNA should be examined, particularly the potential to induce RNA interference in humans. And this can be done through an in silico analysis to identify any homologous RNA in humans, followed up with a literature search to identify homologs. This information, when coupled with the tiered toxicity studies, will inform on the hazard of the RNA being developed. In addition to sequence-specific effects, non-sequence-specific effects should also be considered, and these include immune responses or the potential for toxicity of the final formulated product. Next slide, please. For human health, the potential routes of exposure that EPA evaluates are dietary if the product is to be applied on food crops. And for occupational exposure, bystanders, and residential uses, we additionally consider ocular, inhalation, and dermal exposures. Next slide, please. After the plausible routes of exposure have been identified, the RNA found in the final end product should be further examined to determine if the human health risk assessment can rely on a history of safe exposure to environmental RNA. This means evaluating if any of the potential exposure routes are influenced by the identity of the RNA. So have there been any chemical modifications to the RNA itself? Also, the end product formulation. Has it been formulated in such a way that the stability or uptake of the RNA can be altered through any of the routes of exposure? And is the RNA in the pesticide product as labile in the gastric environment as plant-derived RNA? Rapid digestion greatly reduces the likelihood of exposure. Next slide, please. Now, looking at environmental hazard considerations, because of the way in which RNA interference works, developers have the unique possibility to design their RNA to be as specific as possible to its intended target organism through the use of bioinformatics during the early R&D pipeline, and therefore minimize the potential for effects in non-target organisms, including humans. Bioinformatics helps developers identify the potential spectrum of activity based on the sequence identity of their RNA. This information will guide the suite of necessary non-target organism studies. The spectrum of activity will then be further analyzed with testing of non-target organisms. These are tiered tests, meaning the initial tests start out by feeding high concentrations of RNA to the examined organism. Next slide, please. For environmental exposure, in addition to bioinformatics, knowledge about the susceptibility of specific non-target organisms to systemic RNA uptake is also informative. But if the topically applied RNA is chemically modified or formulated, then developers should address how this could change the spectrum of activity of the product. Next slide, please. The environmental exposure in the field. Let's see. Can we go to the next slide, please? Oh, go back one. There we go. The environmental exposure in the field also needs to be considered. This involves identifying those non-target organisms that could potentially be exposed to the topically applied RNA, such as insects that naturally associate with the crop on which the product is to be applied, and how the product is applied. Is it sprayed or is it applied as a seed treatment? This exposure consideration also looks at the physical properties of the RNA. So typically non-modified RNA degrades very quickly in the environment. And so developers should examine whether this property is changed in the product they're developing, and the degradation profile of that topically applied RNA should also be assessed. Next slide, please. In summary, humans and other non-target organisms have a history of safe exposure to plant-derived RNA. For RNA that is topically applied, the RNA may need to be modified or formulated to stabilize the molecule or to enhance its activity. These changes to the molecule may then result in a change of the hazard or exposure profile for humans and non-target organisms, which needs to be addressed in the pesticide application. Next slide. And with that, I would like to thank you all very much for your time and attention today.
Thank you, Dr. Nina. I appreciate that you really use your time. Actually, you gave us a few minutes.
Yeah.
Thank you.
I tried to.
Thank you. Well, our next speaker is the Master in Science, Alma Liliana Tobar Diaz. She's working for Senacica, Mexico, and her topic will be about pesticides in Mexico, regulation and trends in the use of new technologies. So, Dr. Alma Liliana, it's your turn now. You have everything on your side.
Muy bien, muchas gracias. Voy a compartir mi pantalla. Primeramente agradecerles a Procinorte, a los organizadores, la oportunidad. Me siento muy honrada de compartir este panel con tan excelentes expertos en su tema. Y bueno, realmente mi presentación, denme un segundo, pues va a ser muy básica, muy general. No sé de los asistentes quienes estén familiarizados con nuestro marco regulatorio para plaguicidas en México, que eso incluye tanto los químicos o convencionales, como aquí les llamamos, así como pues los bioplaguicidas, que señalar que, denme un segundo, que no logro poner la pantalla completa. Ay, me estorba el panel de... Listo. Y bueno, en nuestro país no existe, como en otros, una categoría o un grupo de productos tales como los bioplaguicidas, pero sí contamos con otras clasificaciones de plaguicidas que, bueno, podrían enmarcarse dentro de este gran grupo de bioplaguicidas. Pero bueno, primero señalarles que nuestro Servicio Nacional de Sanidad, Inocuidad y Calidad Agroalimentaria pues tiene la misión de proteger los recursos agrícolas, acuícolas y pecuarios de plagas y enfermedades a través de básicamente buenas prácticas agrícolas, pecuarias, acuícolas, pesqueras, para facilitar el comercio nacional e internacional de los bienes que derivan de estos sistemas de producción. El marco jurídico aplicable específicamente a plaguicidas es el que ahorita les estoy mostrando. Tenemos una variedad de leyes sustantivas que nosotros les llamamos. Para el tema de plaguicidas es principalmente la Ley General de Salud, de la cual deriva pues toda la regulación relacionada con los productos, dispositivos, materiales que pudieran tener este impacto en la salud y que tienen que tener diversa serie de autorizaciones sanitarias, tales como permisos, licencias o en el caso de plaguicidas un registro sanitario para poderse utilizar en el país. Y bueno, de la mano de esta, para plaguicidas tenemos en el caso de Senacica, que es el sector de agricultura en el gobierno mexicano, tenemos la Ley Federal de Sanidad Vegetal, que va a regular y promover la sanidad vegetal, las buenas prácticas, así como la efectividad, entre muchas otras cosas, desde luego, de los insumos fitosanitarios y de los métodos de control integrado. Y hay un par de leyes muy importantes en materia ambiental, que son la Ley General para la Prevención y Gestión Integral de los Residuos, que entre otras cosas va a regular lo relacionado con el manejo y disposición de los residuos de plaguicidas, entre otras sustancias tóxicas o peligrosas, y sus envases vacíos, y la Ley General del Equilibrio Ecológico y la Protección al Ambiente, que regula la aplicación de plaguicidas, fertilizantes, sustancias tóxicas, y también otorga autorizaciones para realizar otras actividades relacionadas. Y derivando de estas leyes, que son nuestras leyes marco, tenemos diversos reglamentos y normas oficiales mexicanas, que aquí lo más relevante de mencionar es el eh, reglamento en materia de registro de plaguicidas, que va a describir tanto los requisitos como los procedimientos para el registro de plaguicidas en México. Complementariamente está la norma de etiquetado, que es cuando ya los productos, bueno, dentro del proceso de registro, tienen que cumplir las características de etiquetado respecto a los usos autorizados, las cantidades, qué se va a hacer con los envases, medidas de protección para especies no blanco como son las abejas y otros polinizadores, entre otros aspectos de importancia. Y bueno, aquí nada más por resaltar esta parte de normas oficiales mexicanas, son estándares ya más específicos. Los tres primeros tienen que ver con servicios y actividades fitosanitarias de comercialización, fabricación, importación de plaguicidas, la aplicación aérea de estos, y otras dos normas que están directamente relacionadas con el registro de productos, que es la de efectividad biológica y límites máximos de residuos en campo que se van a utilizar, bueno, para que después el área de salud autorice esos límites máximos dentro del proceso de registro. Entonces, en este registro sanitario es importante tener muy presente que la autoridad que coordina el proceso como tal es la COFEPRIS, la Comisión Federal para la Protección contra Riesgos Sanitarios, que es un órgano de la Secretaría de Salud. Ellos van a hacer toda la evaluación que tiene que ver con que el producto sea seguro en las condiciones en que se va a utilizar en el país, tanto para quienes lo van a manejar o lo van a aplicar, y desde luego a los consumidores. La Secretaría de Medio Ambiente, que es la que va a evaluar todos los riesgos al ambiente, ya sea suelo, agua, organismos no blancos. Y en nuestro caso, agricultura, bueno, Senacica no lo mencioné, es un órgano desconcentrado de la Secretaría de Agricultura, y nosotros tenemos la participación en la evaluación de la efectividad biológica de los productos en campo, porque, bueno, es importante que estos productos, además de ser seguros para la salud humana y el ambiente, tengan este efecto en las plagas y enfermedades de interés. Y aquí abajito les pusimos el nombre del reglamento, es un reglamento con un nombre un poco largo que, bueno, aquí históricamente se le ha conocido como RPLAFES, que actualmente está en un proceso de revisión, precisamente como mencionaré más adelante, pues para tratar de incorporar la regulación de estas nuevas tecnologías que, bueno, en muchos países ya van más avanzados, pero en México estamos un poquito, digamos que un poquito rezagados. Entonces, para ejemplificar el proceso de registro, bueno, COFEPRIS, como ya mencioné, es la autoridad responsable de coordinar este proceso. Ellos van a recibir la solicitud de registro por parte de una empresa interesada en registrar. Este expediente, esta solicitud está conformada de varias partes. Entonces, las partes que corresponden a salud las va a evaluar COFEPRIS, las partes que corresponde evaluar a medio ambiente las va a evaluar esa secretaría, y las partes que corresponden a agricultura las vamos a evaluar Senacica para el caso de plaguicidas, y directamente la Secretaría de Agricultura cuando se trata de nutrientes vegetales. Las tres dependencias van a emitir una opinión. Las tres tienen que ser favorables y son vinculantes, es decir, si dos de las tres o una de las tres emite una opinión no favorable, no puede emitirse ese registro y el solicitante tendrá que atender ya sea los requerimientos de información, o en caso de no cumplimiento, pues este trámite será desechado, y si posteriormente se completa la información, puede solicitar nuevamente ese registro. Una vez que los registros son emitidos, COFEPRIS tiene este portal oficial donde podemos consultar estos registros de plaguicidas emitidos, donde ustedes van a poder conocer el número de registros, la clave de registro del producto, a qué empresa em, sí, corresponde, quién es el titular, el dueño de ese registro, el ingrediente activo y su nombre comercial, a qué categoría toxicológica pertenece. En México tenemos 5 categorías toxicológicas vigentes, los usos autorizados y la vigencia de este registro, que para México es por 5 años. A continuación, solamente para ejemplificar un poquito la evolución en los últimos años de los tipos de plaguicidas que se han estado evaluando y registrando en nuestro país. Bueno, aquí tenemos en la parte de efectividad biológica, que es lo que nos compete, desde el 2018 hasta lo que va de este año. Bueno, ustedes podrán observar el total en la barra azul, en amarillo los convencionales, que son todos los químicos, y en verde los no convencionales, que son todos los demás, incluyendo, ahorita vamos a ver algunas de estas tecnologías novedosas, que en su momento aquí cabrían pues los plaguicidas a base de ARN de interferencia. Esto es por la parte de la efectividad de los productos en campo, y luego por la parte de los registros, Senacica, así como Semarnat, emiten una opinión técnica, donde con base en las atribuciones que ya mencioné, vamos a decir la información correspondiente vaya, cumple con los criterios del reglamento para que ese producto se pueda registrar o no. Y aquí, de acuerdo a lo que COFEPRIS nos manda para evaluar, pues es la misma código de colores, en azul en total del 2018 a lo que llevamos de 2025, en naranja todos los productos químicos. Evidentemente sigue habiendo un mayor desarrollo, un mayor registro de este tipo de productos, y en la barra verde los no convencionales, todos los que no son químicos. Tal vez lo que habría que resaltar de esto es que cada vez más empresas, tanto personas físicas como morales, pues están interesadas en registrar productos que no sean químicos preferentemente, y que además sean menos riesgosos o peligrosos para el ambiente, para la salud, y que sigan teniendo estas características de ser efectivos para el control de diversas plagas y enfermedades. De esta manera, de acuerdo a lo que nosotros como Senacica hemos evaluado en los últimos tal vez 4 años y medio, pues en primer lugar, bueno, sí, digamos que en frecuencia de estas nuevas tecnologías que se han estado revisando, que realmente son muy pocas las solicitudes que para estas tecnologías se han presentado para registro. Pero bueno, tenemos los péptidos, que sabemos que no es, por ahí se mencionó en una de las presentaciones, eh, para usos como fungicidas o insecticidas, el virus de la poliedrosis nuclear para uso como insecticida, y por ahí nada más una o tal vez 2 nanopartículas para uso como fungicida, y todos estos para el control de diversas malezas y plagas en una amplia variedad de cultivos. En cuanto a ARN, de mencionar que en México aún no contamos con, a petición de algún interesado, solicitudes para registrar este tipo de productos a base de esta tecnología. sabemos que hay una persona moral interesada, una empresa interesada en ya traer esa tecnología a México, sin poder dar los detalles porque el proceso, como menciono, lo tiene de su lado la COFEPRIS, pero sabemos que hay un par por ahí o tres productos que de hecho ya tienen registros en Estados Unidos a base de ARNi y que esa empresa tiene interés de traer ya la tecnología y tal vez la producción de ese material aquí a México. Pero esto es algo muy, muy reciente que está ocurriendo, de hecho, en este año. Entonces, para nosotros es muy interesante estar participando ahorita con ustedes porque pues nos estamos enterando de ya todas las experiencias y los retos que ustedes han tenido tanto para evaluar como para en su momento registrar estos productos. Y bueno, desde luego, ¿cuáles son los retos que me parece que para cualquier tecnología siempre se van a tener? En el caso de México, resaltar que, bueno, nos hace falta aún trabajar o contar con una regulación más moderna y más eficiente. Comentarles que desde hace varios meses las autoridades competentes, salud, ambiente, agricultura, economía, la parte esta de comercio, estamos trabajando en una revisión del reglamento de registro, de manera que podamos incorporar pues todos aquellos requisitos y procedimientos necesarios para poder registrar en México y usar adecuadamente estas tecnologías. De la mano de esto, evidentemente, tiene que haber una plantilla de evaluadores más especializados. En todas las dependencias hay personal con perfiles profesionales muy particulares, que a lo largo de los años se han ido especializando cada vez más en la evaluación de los productos. Sin embargo, para estas nuevas tecnologías, pues hará falta prepararse todavía más. Y también un reto, tal vez no puse mucha atención en sus presentaciones, pero un reto que tenemos también aquí como países es mejorar las prácticas de manejo integrado de plagas, dado que muchas de estas nuevas tecnologías que se están viendo, pues tienen que ir dentro de este enfoque de manejo integrado. Y todo esto de manera que se favorezca el menor uso de plaguicidas químicos, pero además aquellos productos que se utilicen contribuyan a adoptar sistemas de producción más sostenibles, efectivos y que contribuyan a minimizar los impactos ambientales que puedan estar generando y desde luego también que puedan estar al alcance de toda la gama de productores en México que tenemos desde los muy pequeñitos hasta productores pues con misión básicamente de exportación. Entonces, pues bueno, esta es la información que les preparé para todos ustedes y a la espera de seguir compartiendo experiencias. Muchas gracias.
Muchas gracias a ti, Alma. Thank you very much to Dr. Alma Liliana. Again, she also very good assignment to her time. So thank you, Doctora. Master in Science, Alma Tovar. The last speaker of today's part of the session is from also the Master in Science, Emily Hopwood. She works for the Health Canada Pest Management Regulatory Agency, and her topic is about regulation of RNAi-based pesticides in Canada. Dr. Emily, there you have it. Go ahead.
Thank you so much. Let me just share my slides as the slideshow. Can you see this okay? And can you also hear me okay?
Perfect.
Okay. Thank you so much. So I'm a senior scientific evaluator with Health Canada's Pest Management Regulatory Agency. And it's a pleasure to be here speaking with all of you today. Thanks for hanging in with me as the last presentation of the day. I'm going to start things off just by giving a high-level overview of how dsRNA pesticides are regulated in Canada. I'm not going to go over how RNAi works since we've talked about that already. And I'm going to focus on describing the different types of data required for dsRNA pesticide registration. Okay, in case anyone hasn't heard of us before, the Pest Management Regulatory Agency is part of Health Canada, and our mandate is to ensure that pesticides have acceptable risk for both human health and the environment, and that they have value. We regulate pesticides throughout their life cycle, so from field research through to registration and then periodic reevaluation. There are some differences in how dsRNA is classified in different jurisdictions around the world. Currently, to the best of my knowledge, the EU is currently regulating topically applied dsRNA as a chemical. In the US, which we just heard about, it falls under their biopesticide framework. And in Canada, it's regulated as a novel class of pesticide. So I'll talk a bit more about that. PMRA's regulatory purview is limited to topically applied dsRNA, so like a spray. Transgenic crops which express pesticidal dsRNAs are regulated by different parts of the government in Canada, so I won't be addressing that. But the most important thing to know about the regulatory framework for dsRNA in Canada is that it's considered a novel class of product, and this is in line with guidance we received from Health Canada's Science Advisory Board back in 2016. And this means that it doesn't fall under any of the existing frameworks for conventional or microbial pesticides, and data requirements are determined case by case. We have experience dealing with dsRNA going back to 2015 when we first started authorizing outdoor research trials. PMRA has been involved in the OECD's expert group on RNAi, along with Nina Ortiz, nice to see you again, and many others from different regulatory jurisdictions. Altogether, we co-authored 2 working documents on dsRNA risk assessment, which are available online. And last year, we published an information note on dsRNA just to communicate to the public like what Canada's current approach is to this. And throughout all of this, we continue to develop our data requirement considerations and provide pre-submission registration advice to potential applicants and assess submissions relating to research trials and whatever else comes our way related to dsRNA. So I'm just skipping over these slides since we have seen a description of the mechanism of action. I'll just briefly pause on this image to remind us all, we've seen similar slides already, but just to remind us that the machinery involved in RNAi feeds into many other cellular processes, and this raises the potential for unintended effects arising from these endogenous systems being perturbed upon application of exogenous RNA. These off-target effects can be dependent on the sequence of the dsRNA, or they can be independent of the sequence of dsRNA, like the interferon response, which is the vertebrate innate immune system activation, which can be triggered by exogenous dsRNA. So with these potential off-target effects in mind, I'll go over the main categories of data required for registration. And just keep in mind that for research authorizations, the requirements are usually reduced compared to what I'm going to talk about, because we realize that these products are at early stages of development and trials are usually small scale. And these 6 review streams are the same for all pesticides, but I'm just going to talk about the dsRNA specific considerations. First of all, for chemistry, we need to see a full description of the manufacturing methods, characterization of the dsRNA itself, as well as any impurities that might be present, and any unintentional ingredients. We need to see a thorough description of any formulants that are combined with the dsRNA to form the end-use product, as well as any chemical modifications that might be made to the dsRNA itself, since all of this kind of informs the hazard profile, which will come next. So under the toxicology stream, excuse me, toxicity, irritation, and sensitization by various routes of exposure will need to be addressed, as well as longer-term impacts like reproductive and developmental effects and carcinogenicity. All of these factors need to be addressed, but not necessarily with an animal study in every case. So some of these requirements can be addressed using rationales based on data from scientific literature and or the results of screening studies. A couple considerations to keep in mind when designing studies to address these data points or in designing waiver rationales is that the study duration might need to be tweaked a little bit. So the exposure and observation periods of in vivo studies might need to be extended, since dsRNA often takes longer to display effects compared to conventional chemical pesticides. And since there's a potential for formulants to affect the properties of dsRNA, like its potential for uptake, the test material may need to be this formulated end-use product instead of the technical active ingredient, depending on the particulars of the product. And then, again, some of the endpoints assessed for, let's say, conventional tox studies might need to be modified for a dsRNA product. So because dsRNA is known to activate the innate immune system, endpoints to assess immunotoxicity might be required depending on like the use pattern of the product and depending on the specific characteristics again. So taking all of this together, it's a very case-by-case approach. And yeah, we just really encourage product developers to get in touch with us early on in the product development process so we can give you tailored advice based on the specific product characteristics. For occupational exposure, the assessment for workers and bystanders is driven by the use pattern, so the application interval, the type of equipment used to apply the product, as well as the results of the tox testing I just mentioned, especially by the inhalation, dermal, and ocular routes. The dietary exposure assessment looks at the amount of residues of the product which may be present on food. This depends on the types of crops proposed for treatment, application timing, whether or not it's proposed for application right up until harvest or not. And we also consider that in vertebrates, there are multiple barriers to dietary uptake of RNA, which have been demonstrated, some of which are listed here. But kind of tempering that is that a lot of the evidence of these barriers comes from studies of unformulated RNA in plants, or from clinical studies which used like modified short interfering RNA to bypass that immune response, which we know that dsRNA can trigger. So while there is a demonstrated history of safe exposure to unmodified RNA from plants, there's not a lot of literature that represents like the concentration of dsRNA to which we could be exposed, and the sort of application type that we could expect from a pesticide. So because of this, empirical oral toxicity studies, as well as residue data, will generally be required to inform a comprehensive dietary risk assessment. For the environmental assessment, data characterizing the environmental fate and behavior of the product will be required, since this will impact the degree to which non-target organisms are exposed. If there are modifications to the dsRNA itself or formulants in the product that could impact its persistence, like some of the examples that we've seen are in development today, the impact of these modifications will need to be addressed on the persistence of the dsRNA in the environment. We require non-target organism studies to address both acute toxicity as well as endpoints like subchronic and chronic toxicity, effects on reproduction and life cycle of non-target organisms. And this is consistent with previous regulatory approaches that Canada has taken in assessing transgenic crops expressing dsRNA, for instance. Which specific test species should be used depends on a couple things, like the use pattern of the product. So, for example, if it's only going to be applied in greenhouses, probably don't need to test avian species, whereas if it's a seed treatment, we would definitely want to see a non-target organism study on birds. Bioinformatics can inform hazard prediction of which non-target organisms might be more susceptible, but wherever possible, we want to see this supported by bioassays. Lastly, Canada requires a value assessment for potential pesticides. So this is an efficacy demonstration. So we need to see that the product generally works on its target pest. And this data is also important because it also informs, like, so the time needed to achieve efficacy also informs what an adequate exposure period and endpoint selection might be for tox and non-target organism testing. And then information on resistance management will also be required since resistance to dsRNA in some insect species has already been documented. I won't get into the details here, but I've listed a couple examples. Okay, this is just to summarize the data requirement considerations on a high level. This is broadly what would be required for each review stream. And then the data from each of these streams is analyzed by many different evaluators and feeds into a comprehensive risk assessment that will determine whether or not the product can be registered. So the main takeaway I hope you'll get from this presentation is that dsRNA is regulated as a novel class of pesticide in Canada. So data requirements are determined case by case. Research authorizations are required for all trials outside of a lab. And we encourage product developers to get in touch with us early in the process so we can give you tailored advice on what's required. And for more information, you can scan this QR code and it'll take you to the information note that we published, which also includes links to the OECD documents on dsRNA risk assessment. So thank you very much for your attention. Here are some references, and I will turn it back to the facilitator.
Wow, thank you, Dr. Emily. You also, were on time for your presentation. I appreciate actually the three speakers from this legislation session. They are all of them use their allotted time. Thank you so much. And because we're running out of time, but still we have a few minutes for questions. Let me see. We have some. Oh, chat. No.
Yeah, I think we have one question, Hemi, while you're looking, but I think, and I know our panelists have answered it to, but I don't think it went to everybody. So I'll read it again, and then maybe I'll ask our the regulatory panelist to provide some insights. So I think the first question is like, how does the regulation changes when the double-stranded RNA is covered on nanoparticles, or maybe applied on the nanoparticle, or applied as nanoparticles? So that's the first question, and then maybe any of the speakers could answer. And then also there's a question about does either country permit use of data from other country for evidence and approvals in registration process? So I know like this has been answered in the chat, but the chat went to the panelists only, but it didn't go to everybody. So if our panelists could just briefly mention about that as well. So Emily or Nina, our speaker from Almatovar. Yeah, go ahead, Emily.
I can take a stab at answering the question about the nanoparticle formulation. I think it would, like, the regulations themselves don't change, but the way in which the data requirements would need to be addressed could potentially change. So for instance, if the dsRNA is unmodified, it may be more possible to rely on scientific literature to address the data requirements for, let's say, like reproductive and developmental toxicity, or like a 90-day oral toxicity study could probably be addressed by scientific literature. But if the dsRNA is encapsulated somehow such that it is it's persistent, or it enters cells in different ways, we'd probably want to see like an in vivo study for a 90-day oral toxicity study in that case. So I hope that that answers the question.
Perfect. No, thank you so much, Emily. And like Camino, for the answer to the question about the data in other countries, maybe you could post it in the chat, the answer provided by US and Mexico. So with that, I think we are at the end of our today's very productive and very the great workshop. And I must thank all of our speakers for their insightful presentation, and to all the participants for the engagement during this session. And while we close this session, I also ask that all of our panelists, if you could start your camera, because ICA, they wanted to have the screenshot of our presenters. So if you could do that, that would be great. And during this session, as you know, like we have explored both the promises and the complexity of RNAi technology for plant health. And we did talk about the fundamental research to some of the practical applications. And it is clear that, the approach holds tremendous potential for advancing pest management. As Dr. Hunter said, the door is open. I think we do have, like the first one in US, which is already registered. And also, I think what stands out from today's discussion is the value of collaboration. By working together, sharing our research, aligning our strategies, and supporting innovation, I think we can accelerate, and we can maybe further accelerate the effective use of RNAi technologies across the globe. And as Dr. Tankos mentioned, like there was a workshop between US and Australia last year. I think we have this workshop as well. So hopefully, I think this would help us to further enhance that collaboration. And in the end, I again would like to thank you, the ProSynode Plant Health Task Force, my colleagues Jaime and Yasmin, and our ICA team, JC, and everyone for organizing this session. And many thanks to our speakers and participants. And then we will come back with more topics of interest to you. And but until then, thank you very much, and enjoy your day.
Yeah. Good.